హలో అండి, గుడ్ ఈవినింగ్. సో ఐ నో లాస్ట్ ఇయర్ ఈ టైంకి మనందరం కాస్త సాడ్ ఫేసెస్ తోటి, కాస్త ప్యానిక్ ఫేసెస్ తోటి, కాస్త భయంగా ఏంటి పేపర్ ఇలా వచ్చేసింది అనే స్టేజ్లో ఉన్నాం. ఐ థింక్ ఈరోజు మోస్ట్ ఆఫ్ ద స్టూడెంట్స్ మేబీ హ్యాపీగా ఉంటారు. డెఫినెట్లీ పేపర్ మీరు అనుకున్నట్లు వచ్చిందని నేను అనుకుంటున్నాను. అండ్ మీకు ఒక ప్రాపర్ అనాలిసిస్ ఒకటి ఇస్తాం, యాజ్ వెల్ యాజ్ ఆన్సర్ కీని. మీ ఫేవరెట్ టీచర్స్ అందరూ ఈరోజు మీకు ఆన్సర్ కీని కూడా మీ ముందు డిస్ప్లే చేస్తారు విత్ ప్రాపర్ ఎక్స్ప్లెనేషన్స్. అయితే ఈరోజు నాతో పాటు రవికాంత్ సార్, మనకు పోరంకి గాయత్రి మిసల్ ఏదైతే ఉందో దాని ఎగ్జిక్యూటివ్ డీన్ సార్, అలాగే నేను మీ గాంధీ సార్. సో అసలు ఫస్ట్ నేను పేరెంట్స్కి స్టూడెంట్స్కి అందరికీ ఒక బిగ్ కంగ్రాచులేషన్స్ చెప్పాలి. ఎందుకు అంటే ఈ జర్నీలో మీరు ఇక్కడ దాకా వచ్చి మీరు నమ్మి, మిమ్మల్ని మీరు నమ్ముకొని, మీరున్నటువంటి కోచింగ్ నమ్ముకొని, మీ కష్టాన్ని నమ్ముకొని ఇక్కడ దాకా వచ్చి మీరు ఎగ్జామ్ని చక్కగా రాసినందుకు మీ గట్స్కి, మీ ఎఫర్ట్స్కి వెరీ 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 బిగ్ కంగ్రాచులేషన్స్. ఎందుకంటే ఇట్ ఈస్ నాట్ హౌ బెస్ట్ యూ స్టార్ట్ ఏ రేస్ ఈజ్ ఇంపార్టెంట్. ఒక రేస్ని ఎంత బాగా స్టార్ట్ చేసామన్న దానికంటే ఒక రేస్ని ఎంత బాగా ఎండ్ చేసామన్నదే ఇంపార్టెంట్. బాగా స్టార్ట్ చేస్తే మెడల్ రాదండి, బాగా ఎండ్ చేస్తే మెడల్ వస్తుంది. కాబట్టి బాగా ఎండ్ చేస్తున్న ప్రతి పిల్లలకి, అండ్ మోస్ట్ ఆఫ్ ద స్టూడెంట్స్ మైట్ హ్యావ్ డెఫినెట్లీ డన్ ఇట్ వెరీ వెరీ వెల్ అండి. అయితే చాలామంది పిల్లలు ఈరోజు నా మాటలతో కొంచెం ఏకీభవిస్తారు. శ్రీ చైతన్యలో చదివిన పిల్లలు చాలామందికి గ్రాండ్ టెస్ట్ పేపర్స్ రాస్తున్నప్పుడు పార్ట్ టెస్ట్లు, అంటే January 19 నుంచి మీకు స్టార్ట్ అయిన రివిజన్స్ టెస్టులు కానివ్వండి, పార్ట్ టెస్టులు కానివ్వండి, స్పెషల్ గ్రాండ్ టెస్టులు కానివ్వండి, గ్రాండ్ టెస్టులు కానీ రాసిన ప్రతిసారి మీకు బయాలజీ టైం కన్జ్యూమింగ్ వచ్చిన ప్రతిసారి మీరు చెప్పిన ఒకే ఒక మాట ఏంటంటే, సార్ బయాలజీ ఇన్ని మల్టీ స్టేట్మెంట్ క్వశ్చన్స్ వస్తున్నాయి, ఎక్కువ టైం కన్జ్యూమ్ చేస్తుంది. మేము రిపీటెడ్గా మీకు చెప్పిన మాట ఏంటి అంటే, ఖచ్చితంగా బయాలజీ మల్టీ స్టేట్మెంట్ క్వశ్చన్స్ మీద మీరు ఫోకస్ చేయాలి. ఈ సంవత్సరం ఎట్టి పరిస్థితుల్లో మీకు బయాలజీ కాస్త ఈజీ సైడ్లో ఉన్న లెంగ్దీ సైడ్లో ఉంటుంది. అంటే లాస్ట్ టైం కూడా నేను చెప్పాను లెంగ్దీ అండ్ డిఫికల్ట్కి డిఫరెన్స్ ఏంటో. లెంగ్దీ టైం కన్జ్యూమింగ్ బట్ స్కోరింగ్. అండ్ ఈసారి స్టేట్మెంట్స్ చాలా ఎక్కువ వచ్చాయి. మనం బయాలజీలో చూస్తే మల్టీ స్టేట్మెంట్ క్వశ్చన్స్ మాత్రమే 23 క్వశ్చన్స్ ఉన్నాయండి, మల్టీ స్టేట్మెంట్స్. బట్ లక్కీలీ ఆ 23 క్వశ్చన్స్ అనేవి టైం కన్జ్యూమింగ్ అయినప్పటికీ మీ అందరికీ డైరెక్ట్ ఎన్సీఆర్టీ లైన్స్ అండ్ ఇబ్బంది పడకుండా, ఇబ్బందులు లేకుండా ఉన్నాయి. కాబట్టి పిల్లలు ఎక్కడా కూడా ఇబ్బంది పడలేదు వీటితోటి. వెరీ వెరీ గుడ్ థింగ్. సెకండ్ ఇంపార్టెంట్ పాయింట్ మనం మాట్లాడుకోవాల్సింది, ఈసారి అసర్షన్ రీజన్ టైప్ ఆఫ్ క్వశ్చన్స్ 0. మనకు బయాలజీ నుంచి అసర్షన్ రీజనింగ్ టైప్ క్వశ్చన్ లేదండి అసలు. ఇది పెద్ద సర్ప్రైజ్ యాక్చువల్లీ. లాస్ట్ ఇయర్ మనకు గుడ్ నెంబర్ ఉంది. సో ఆ నెంబర్ని ఏం చేశారంటే మల్టీ స్టేట్మెంట్ యాజ్ వెల్ యాజ్ మ్యాచ్ ద ఫాలోయింగ్లో కన్వర్ట్ చేశారు. అండ్ మ్యాచ్ ద ఫాలోయింగ్ కూడా మనం ఈసారి పేపర్ని బయాలజీ మ్యాచ్ ద ఫాలోయింగ్ చూస్తే 99.99% 2024 పేపర్ మీద ఏదైతే చూసారో 2024 బయాలజీలో మ్యాచ్ ద ఫాలోయింగ్ కనీసం అంటే వన్ ఆఫ్ ది ఆప్షన్స్ ఇఫ్ యూ చూస్ ఇట్ విల్ బి రైట్. అంటే A3 అనుకోండి, మీరు చూస్తే A3 ఒక దాంట్లోనే ఉంది కరెక్ట్. అంటే టైం కన్జ్యూమింగ్ కాదు. బట్ మల్టీ స్టేట్మెంట్ క్వశ్చన్స్లో దో ఎన్సీఆర్టీ లైన్స్ నుంచి డైరెక్ట్గా క్వశ్చన్స్ వచ్చినప్పటికీ కాస్త టైం కన్జ్యూమ్ అవుతుంది. అంటే ఇక్కడ మనము చాలా జాగ్రత్తగా ఆలోచించాల్సిన ఒక విషయం ఉందండి. ఇంటర్మీడియట్ పిల్లల్ని, లాంగ్ టర్మ్ పిల్లల్ని ఒకసారి మనం కాస్త ఇక్కడ వేరియేట్ చేసి మాట్లాడాలి. ఎందుకు అంటే ఇప్పుడు ఇంటర్మీడియట్ నుంచి వెళ్ళిన పిల్లలకు జనరల్గా ఏమవుతుందంటే ఈ పేపర్లో బయాలజీని ఎంతో కొంత కాస్త లెంగ్తీ ఎక్కువ అనిపించవచ్చు. ఎందుకంటే లాంగ్ టర్మ్ పిల్లలు బయాలజీ చాలా బాగా చేస్తారండి. అంటే నేను బాగా మెరిట్ అనుకోండి, ఈ పేపర్ని ఒక 50 minutesలో బయాలజీని కంప్లీట్ చేస్తారండి. అదే లాంగ్ టర్మ్ పిల్లలు అనుకోండి, డీసెంట్గా 1 hourలో ఈ పేపర్ని కంప్లీట్ చేసి ఉండొచ్చు. సో మీరు కనుక 1 hour టైం తీసుకొని ఈ పేపర్ని కంప్లీట్ చేసి ఉంటే బయాలజీ డెఫినెట్లీ మీరు మంచి టైం ఇచ్చుంటారు. అండ్ అంబిగ్వియస్ క్వశ్చన్స్ కానీ, లేదు అంటే బాగా కన్ఫ్యూజింగ్ క్వశ్చన్స్ కానీ మనకు బయాలజీలో ఎక్కడా కనిపించలేదు. కాబట్టి బయాలజీ 360/360 వచ్చే అవకాశాలు చాలా ఎక్కువ కనబడుతున్నాయి. గుడ్ థింగ్ ఏంటంటే చాలామంది లాంగ్ టర్మ్ పిల్లలు ఇప్పుడు కాల్ చేసి చెప్తున్న మాట కూడా ఏంటంటే, సార్ చాలా బాగా చేశాము, మన గ్రాండ్ టెస్ట్ పేపర్స్ చాలా హెల్ప్ అయ్యాయి సార్, ఆల్మోస్ట్ టఫెస్ట్ పేపర్స్ అని రాసేసాము. అండ్ ఈ పేపర్ మీరు లాస్ట్ టైం కూడా నేను చెప్పాను, నానా 24 కంటే ఎక్కువ, 25 కంటే తక్కువ పేపర్ రావచ్చు, బట్ మీరు టఫ్ పేపర్స్ కూడా రెడీ అవ్వండి అని. మీరు టఫ్ పేపర్స్కి రెడీ అయ్యారు, మోడరేట్ పేపర్స్కి రెడీ అయ్యారు, ఈజీ పేపర్లకి రెడీ అయ్యారు. మిమ్మల్ని డిఫరెంట్ కైండ్ ఆఫ్ ఎగ్జామ్స్కి రెడీ చేసి దగ్గర ఈ ఇయర్లో మీరు 102 పేపర్స్ రాశారండి పిల్లల ఎగ్జామ్స్. ఈ 102 పేపర్ల రిజల్ట్ ఏంటనేది మీకు టీచర్స్ కాసేపట్లో అనౌన్స్ చేస్తారన్నమాట. అంటే ఎలా క్వశ్చన్స్ డైరెక్ట్గా క్లాస్ రూమ్ టీచింగ్ నుంచి కానివ్వండి, డైరెక్ట్గా క్వశ్చన్ పేపర్స్ నుంచి, అంటే మనం ఇచ్చిన క్వశ్చన్ పేపర్స్ నుంచి 90-95% ఆఫ్ ది క్వశ్చన్స్ మనకు డైరెక్ట్గా ఎగ్జామ్లో మ్యాచ్ అవుతున్నాయి. అంటే దట్ ఈస్ ది స్ట్రెంగ్త్ ఆఫ్ శ్రీ చైతన్య కరికులం కానివ్వండి, మన క్వశ్చన్ పేపర్ స్ట్రెంగ్త్ కానివ్వండి. అంటే ఒక ప్రాపర్ R&D అనేది ఎప్పుడు అవసరం అండి. జస్ట్ లైక్ దట్ మనం వెళ్ళకూడదు. అయితే బయాలజీ విషయానికి వస్తే మనం డైరెక్ట్గా చెప్పాల్సింది ఏంటంటే బయాలజీ వాజ్ లిటిల్ టైమ్ టేకింగ్ బట్ స్కోరబుల్. 360 ఈజ్ పాసిబుల్. మీరు సీట్ గెట్టింగ్లోకి వెళ్ళాలంటే 330+ ఖచ్చితంగా మీకు అవసరమనే అనిపిస్తుంది బయాలజీలో ఓపెన్ కేటగిరీ పిల్లలకి. అండ్ రిమైనింగ్ వాటి గురించి మనం ఒక 1 వీక్ టైం తీసుకొని ఆల్ ఇండియా డేటా అంతా అనాలిసిస్ చేసుకొని దాని బేసిస్ మీద మీకు ప్రాపర్ ప్రెడిక్షన్స్ ఏంటి అని మాట్లాడుకుందాం. బికాస్ దిస్ ఈజ్ వెరీ ప్రిమిటివ్ టైం. మనం ప్రీమెచ్యూర్ ఎప్పుడు కూడా డిస్కషన్స్ చేయకూడదండి. వీ షుడ్ ఆల్వేస్ హ్యావ్ మెచ్యూర్ డిస్కషన్స్. కాబట్టి అలాగే పిల్లలకు కూడా ఏంటంటే అన్వాంటెడ్ ట్రామా స్ట్రెస్ మనం పెట్టకూడదు. అండ్ నెక్స్ట్ మనము కెమిస్ట్రీకి వస్తే, కెమిస్ట్రీలో ఫిజికల్ కెమిస్ట్రీ నుంచి 18 క్వశ్చన్స్, ఆర్గానిక్ కెమిస్ట్రీ నుంచి మనకు 14 క్వశ్చన్స్, అలాగే ఇనార్గానిక్ కెమిస్ట్రీ నుంచి 13 క్వశ్చన్స్ రావడం జరిగిందండి. ఈ ఇనార్గానిక్లో ఉన్న 13 క్వశ్చన్స్ డైరెక్ట్ క్వశ్చన్స్, ఎన్సీఆర్టీ లైన్స్, ఇబ్బంది లేదు. ఆర్గానిక్ కెమిస్ట్రీలో మనకు మీరు కనుక కోడ్ 12లో ఉంటే కనుక క్వశ్చన్ నెంబర్ 72 ఒకటి మనకు 3, 4 రెండు ఆప్షన్స్ వస్తున్నాయండి. సో ఆ ఒక క్వశ్చన్ తీసేస్తే మిగిలినవన్నీ కూడా ఆల్మోస్ట్ డైరెక్ట్ క్వశ్చన్స్, సేఫ్ క్వశ్చన్స్, అంబిగ్విటీ కూడా ఎక్కడ లేదు. ఈజీలీ డూయబుల్ పేపర్ అండి. వేర్ యాజ్ ఫిజికల్ కెమిస్ట్రీలోకి వస్తే ఎలక్ట్రోకెమిస్ట్రీలోంచి అలాగే థర్మోడైనమిక్స్ నుంచి ఒక 2-3 క్వశ్చన్స్ కాస్త ఎక్సెసివ్ క్యాలిక్యులేటివ్ ఉన్నాయి. అంటే ఫిజికల్ కెమిస్ట్రీ కాస్త టైం కన్జ్యూమింగ్ అనిపించింది. క్యాలిక్యులేటివ్ పార్ట్ మనకు నన్నస్ట్ ఈక్వేషన్ మీద వచ్చినటువంటి ఎలక్ట్రోకెమిస్ట్రీ క్వశ్చన్ కానివ్వండి, థర్మోడైనమిక్స్ నుంచి వచ్చిన క్వశ్చన్ కానివ్వండి, కాస్త టైం టేకింగ్ అనిపించింది పిల్లలకి. అక్కడ కాస్త టైం తీసుకున్నారు. బట్ ఇట్స్ ఎ వెరీ గుడ్ అండ్ డ్యూయబుల్ పేపర్. మీరు 55 మినిట్స్ నుంచి 1 అవర్ మేబీ కెమిస్ట్రీకి ఇచ్చి ఉంటే కనుక, అలాగే మీరు ప్రాపర్గా చేస్తుంటే ఈ పేపర్లో 35 క్వశ్చన్స్ కనుక ఒకవేళ చేస్తుంటే మీ ఆప్షన్స్, మీ అవకాశాలు చాలా 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 మెరుగుపడతాయండి. సో 330 మార్క్స్ ఇన్ బయాలజీ అండ్ ఇక్కడ ఒక 35 క్వశ్చన్స్ మీరు చేస్తుంటే చేయగలిగిన క్వశ్చన్స్ 35 ఈజీ కనబడుతున్నాయి. కాబట్టి డెఫినెట్లీ ఇది ఎక్కడో మీకు ఒక ఎడ్జ్ వస్తుందండి. అలాగే మన ఫిజిక్స్కి వస్తే కనుక, ఫిజిక్స్ మనము లాస్ట్ ఇయర్ పేపర్ చూసిన తర్వాత చాలా భయపడిపోయారు పిల్లలు. ఈ సంవత్సరం కూడా అలా వస్తుందేమో అని ఇబ్బంది పడ్డారు. బట్ ఫిజిక్స్ ఈజ్ సర్ప్రైజింగ్. ఎన్టీఏ ఈజ్ ఆల్వేస్ సర్ప్రైజింగ్. ఎప్పుడు అంటే తన వైపు నుంచి వచ్చే ఒక సర్ప్రైజ్. 2024 పేపర్ వాజ్ వెరీ ఈజీ, సర్ప్రైజ్డ్. 2025 పేపర్ వాజ్ వెరీ టఫ్, సర్ప్రైజ్డ్. అండ్ దెన్ ఎన్టీఏ ట్వీట్స్. కరెక్ట్గా ఎగ్జామ్కి ఒక 1 వీక్ ముందు ఎన్టీఏ ట్వీట్స్. ఇట్ ఈస్ నాట్ జస్ట్ లైక్ అన్ అదర్ ఎగ్జామినేషన్. ఆ రోజు కూడా నేను చెప్పాను మీకు, వెరీ క్లియర్లీ ఎక్స్ప్లెయిన్ చేశాను పేరెంట్స్ అందరికీ. ఎన్టీఏ ఇక్కడ నేను టఫ్కి ఇస్తాను, ఈజీకి ఇస్తున్నాను, మోడరేట్కి ఇస్తున్నాను ఎక్కడ కూడా మెన్షన్ చేయట్లేదు. మీ రెడీనెస్ గురించి మాట్లాడుతుంది, మీ టైం మేనేజ్మెంట్ స్కిల్స్ గురించి మాట్లాడుతుంది. ఎలాంటి పేపర్ వచ్చినా మేము ఫిల్టర్ చేస్తాము అనే దాని గురించి మాట్లాడింది. అండ్ మనం దాని గురించి చాలా క్లియర్గా ఒక లైవ్ సెషన్ పెట్టుకొని డిస్కస్ చేసాము. ఐ థింక్ సార్ ఇట్ ఈస్ వెరీ నెసెసరీ. ఎందుకంటే పిల్లలకు పేరెంట్స్కి మధ్యలో ఉన్నటువంటి ఆ చిన్న క్వశ్చన్స్ ఏవైతే మైండ్లో ఉంటాయో వాటిని ఎప్పుడు మనం రిజాల్వ్ చేయాలండి. అది మేము రిజాల్వ్ చేయడంలో సక్సెస్ఫుల్ అయ్యాము. పిల్లలకు కానివ్వండి అనికి కౌన్సిలింగ్ సెషన్స్ పెట్టుకొని చెప్పుకోగలిగాము. కాబట్టి పిల్లలందరూ కూడా వాటి కోసం రెడీ ఉన్నారు. ఐఎమ్ వెరీ వెరీ హ్యాపీ. ఫిజిక్స్ పేపర్కి వస్తే కనుక ఫిజిక్స్ క్యాలిక్యులేటివ్ పార్ట్ చాలా ఎక్కువ ఉంది. అంటే న్యూమెరికల్స్ మీద ఎక్కువ బేసిస్, జనరల్గా ఎక్కువ న్యూమెరికల్ బేస్డ్ పేపర్ వచ్చింది. ఫార్ములా బేస్డ్ పేపర్ ఎక్కువ ఇబ్బంది పెట్టలేదు. ఒక కొన్ని క్వశ్చన్స్ 4-5 క్వశ్చన్స్ కొంచెం క్యాలిక్యులేటివ్ క్వశ్చన్స్ ఉన్నాయి. అదర్వైజ్ ఫిజిక్స్ వాజ్ ఎ వెరీ 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 ఈజీ పేపర్. అండ్ కంపేరిటివ్ టు 25 అయితే చాలా ఈజీ పేపర్, 24 కంటే కాస్త బెటర్ పేపర్. సో వీ కెన్ సే దట్ లెంగ్దీ బట్ డూయబుల్ పేపర్. సో స్కోరింగ్ పేపర్ అంటే ఫిజిక్స్లో కూడా మీరు 25 నుంచి 30 క్వశ్చన్స్ కనుక చక్కగా చేయగలిగి ఉంటే మీ యొక్క సెలక్షన్ ఛాన్సెస్ ఆర్ వెరీ 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 హై. సో ఫిజిక్స్ కంప్లీట్లీ అప్లికేషన్ బేస్డ్ చాలా చాలా తక్కువ అప్లికేషన్ బేస్డ్ ఉన్నాయి, ఎక్కువ న్యూమెరికల్ బేస్డ్ క్వశ్చన్స్ వచ్చాయి. వేరస్ కెమిస్ట్రీ దెర్ వర్ వెరీ గుడ్ క్వశ్చన్స్ వేర్ అప్లికేషన్ బేస్డ్ క్వశ్చన్స్ వర్ దేర్. ఓవరాల్గా చూస్తే మనకు ఫిజిక్స్ కాస్త లెంగ్దీగా ఉన్న, కెమిస్ట్రీ కొంచెం లెంగ్దీ అండ్ ట్రిక్కీగా ఉన్న, బయాలజీ వాజ్ లెంగ్దీ బట్ ఈజీ. ఓవరాల్ పేపర్ ఈజ్ మోడరేట్ అని చెప్పుకోవచ్చు అండి మనం. ఈజీ టు మోడరేట్, నాట్ టు మోడరేట్ టు లెంగ్దీ ఆర్ మోడరేట్ టు డిఫికల్ట్ కాదు, బట్ ఈజీ టు మోడరేట్ పేపర్. కాబట్టి కట్ ఆఫ్స్ లాస్ట్ ఇయర్ కంటే కాస్త స్పైక్ అయ్యే ఛాన్స్ ఉన్నాయి. మరీ ఎక్కువగా 24 కట్ ఆఫ్స్ కాకపోయినా బట్ కట్ ఆఫ్స్ అనేది స్పైక్ అయ్యే ఛాన్స్ అయితే వెరీ 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 హై అండి. నవ్ ఐ వాంట్ మన రవికాంత్ గారు టు హ్యావ్ ఫ్యూ వర్డ్స్ అండి.
యా, సో ఓవరాల్ మనకి లాస్ట్ ఇయర్ పిల్లలు ఎగ్జామ్ రాసినప్పుడు చాలా కంగారుగా ఆల్మోస్ట్ 2.5 hours they were really stressed. But this time I am so happy that after maybe for 5-10 minutes, after 5-10 minutes they would have understood that the paper is not so difficult, or maybe so they were so relaxed. I am sure the children moral would have boosted very much, and they would have done really well. And one more good thing is, especially for the Sri Chaitanya children, lastly we have conducted what are the grand tests. Almost all the questions, you can say almost 70 to 80% of the questions were almost similar.
Yes,
like say even semiconductors, modern physics and physics especially. So children after seeing those questions definitely వాళ్ళు చాలా హ్యాపీగా రిలాక్స్డ్ గా అనిపించుంటుంది. ఎందుకంటే ఎస్పెషల్లీ ఫిజిక్స్ అంటే కొంచెం కంగారుగా ఎలా వస్తుంది పేపర్ అని టెన్షన్ లో ఉంటారు. బట్ పిల్లలు అయితే డెఫినెట్ గా చాలా హ్యాపీగా ఫీల్ అయి ఉంటారు. అండ్ డెఫినెట్గా స్కోర్స్ అయితే యాజ్ ఇనీషియల్ మీరు చెప్పినట్టుగా కట్ ఆఫ్ కొంచెం పెరగచ్చు. బట్ మంచి పిల్లలు, బాగా డిసిప్లిన్డ్గా ప్రిపేర్ అయిన పిల్లలు డెఫినెట్గా మంచిగా క్వాలిఫై అవుతారు, డాక్టర్స్ అవుతారు. సో పేపర్ ఈస్ లిటిల్ స్టూడెంట్ ఫ్రెండ్లీ. లాస్ట్ టైం ఇట్ ఈస్ నాట్ అట్ ఆల్ స్టూడెంట్ ఫ్రెండ్లీ. ఇట్ ఇస్ సో పిల్లలు చాలా యాంగ్షియస్గా, చాలా కంగారు పడి 3 గంటలు ఆల్మోస్ట్ 2 గంటలు వేస్ట్ చేసిన పిల్లలు కూడా ఉన్నారు.
కరెక్ట్.
దిస్ టైమ్ దే వుడ్ హ్యావ్ యూజ్డ్ ఆల్ 3 hours effectively to answer the paper.
Correct.
So I'm happy that NTA has conducted some, earlier regarding the tweet, what Gandhi sir is saying. Tweet is not at all regarding the toughness of the paper, if you really understand. It is about how they are prepared, So every center, today we have visited few centers and we have got reports from different centers. Andru cheppedani chala organized ga chesarani cheppi. So NTA's tweet is fine. Like organized ga chala proper ga ready ga unnaru NTA vallu. అదే ట్వీట్లో చేశారు. కాకపోతే పిల్లలు ఎంత కాదన్నా సెన్సిటివ్గా ఉంటారు కాబట్టి ఇది రిలేటెడ్ టు పేపర్ అనుకొని కొంచెం కంగారు పడ్డారు. బట్ నౌ పేపర్ ఈజ్ రియలీ సర్ప్రైజింగ్
బికాస్ మోస్ట్ ఆఫ్ ది సోషల్ మీడియా స్టంట్ వాళ్ళు ఏంటంటే పిల్లల్ని ఎప్పుడు కూడా ఎక్కువ యాంగ్జైటీ క్రియేట్ చేద్దాము, యాంగ్జైటీ తీసుకొద్దాము అనే ఆలోచనలో ఉంటారండి. బట్ యాజ్ ఐ రైట్లీ టోల్డ్ బిఫోర్ ఎగ్జామ్ ఆల్సో అండి, మీ అందరికీ ఎగ్జామ్ అనేది కామన్ పేపర్. మోడరేట్గా వచ్చింది అందరికీ, మోడరేట్గా వచ్చింది. ఈజీ వచ్చింటే అందరికీ ఈజీ, టఫ్ వచ్చింటే అందరికీ టఫ్.
ట్రూ.
సో ఇట్స్ ఎ రిలేటివ్ పేపర్. మనం ఎక్కడ ఇబ్బంది పడాల్సిన అవసరం లేదు. బట్ 3 గంటలు నిజంగా మీరు సిన్సియర్గా ఎఫర్ట్ పెట్టి మీరు చక్కగా రాసి ఉంటే ఇట్ ఈస్ వెరీ వెరీ గుడ్. నాకు అంతటికంటే బాగా అనిపించింది ఏంటంటే సార్, ఈరోజు ఫస్ట్ టైం విజయవాడలో చాలా రోజుల తర్వాత క్లైమేట్ చల్లగా ఉంది. నేను ఎన్నిసార్లు అనుకున్నానంటే పిల్లలు ఎగ్జామ్ చక్కగా రాస్తున్నారు, మరీ ఎక్కువ ఎండలేదు, చక్కగా గాలి కూడా వస్తుంది, హ్యాపీగా అనిపించింది. పాపం పేరెంట్స్ కూడా 3 hours బయట వెయిట్ చేయాలి. అండ్ కొన్ని సెంటర్స్లో అయితే ఐఎమ్ వెరీ వెరీ హ్యాపీ అండి. ఆ సెంటర్స్లో పిల్లలకి పేరెంట్స్ కోసం టెంట్స్ అవి అరేంజ్ చేశారు అండ్ జనరేటర్స్ పెట్టారు. అండ్ ఇంకా ఇంపార్టెంట్ థింగ్ ఏంటంటే ఎన్టీఏ హ్యావ్ టేకెన్ ఆల్ పాసిబుల్ ఫెసిలిటీస్ అండి. దీనికి ముఖ్యంగా ఆంధ్రప్రదేశ్ మనం రాష్ట్రంలో కానీ తెలంగాణలో కానీ చూస్తే ఇక్కడ ఏపీ అసోసియేషన్స్, పేరెంట్స్ అసోసియేషన్స్ ఏవైతే ఉన్నాయో వాళ్ళందరూ కూడా ఎన్ని అసోసియేషన్స్ ఉన్నాయో అన్ని, అది అరుణ్ చౌదరి గారు కానివ్వండి, శ్రీనివాస్ గారు కానివ్వండి, బాబ్జీ గారు కానివ్వండి, వీళ్ళందరూ డిఫరెంట్ పేరెంట్స్ తోటి మీరు పడ్డ కష్టం, మీరు తిరిగిన విధానము, మీరు డిఫరెంట్ పీపుల్తో కలవడము, అలాగే డిఫరెంట్ ఆర్గనైజేషన్స్ నుంచి కూడా సపోర్ట్ రావడము, డిఫరెంట్ కోచింగ్ ఇన్స్టిట్యూట్స్ నుంచి కూడా సపోర్ట్ దొరకడము, అందరూ ఒకటైపోయి పిల్లల కోసం కష్టపడ్డారండి. కాబట్టి ఇది ఒక యునైటెడ్ కలెక్టివ్ ఎఫర్ట్. ఇలానే అందరం కలిసి మన పిల్లల కోసం, మన పిల్లల భవిష్యత్తు కోసం వర్క్ చేయాలి. అండ్ వితౌట్ ఎనీ డిలే ఐఎమ్ సీయింగ్ ఆల్ ద పీపుల్ ఆర్ స్పామింగ్. నేను కూడా ఎక్సైటెడ్, ఎందుకంటే చాలా మంది స్టూడెంట్స్ ఫోన్స్ వస్తున్నాయి, రిపీటెడ్గా మా ఆపరేటర్ చూపిస్తున్నారు నాకు, సార్ కాల్స్ వస్తున్నాయని. అండ్ ఆల్రెడీ ఒక 20-30 కాల్స్ మాట్లాడాను. వెరీ వెరీ హ్యాపీ. ఆల్ ద స్కోర్స్ ఆర్ కమింగ్ వెరీ గుడ్ అండి. అండ్ వితౌట్ ఎనీ డిలే we'll start with the biology. Yeah, please sir.
అందరూ కట్ ఆఫ్ ఎంత ఉంటుంది అని అడుగుతున్నారు. సో ప్లీజ్ రిలాక్స్ అట్లీస్ట్ ఫర్ 1 or 2 days and after that let us think about this cut off and all that. ఎంతో కష్టపడి 1 year and 2 years, చాలా మంది 3 years కూడా కష్టపడి ఉంటారు. and this is your time. exam is over now. సో కొంచెం ప్రశాంతంగా ఇంటికి వెళ్ళి కొంచెం రెస్ట్ తీసుకోండి, మంచి రెస్టారెంట్కి వెళ్ళి మంచిగా భోజనం చేయండి, ఇంట్లో వాళ్ళతో మాట్లాడండి, సినిమా చూడండి, ఏదైనా మిగిలితే
ఎవ్రీ వీక్ సినిమా సినిమా చూసేయండి, పేరెంట్స్ కి టైం ఇవ్వండి. అండ్ ఈ అనాలిసిస్ ఇవన్నీ వీటి మీద ఎక్కువ డిపెండ్ కాకండి అమ్మా. వి విల్ గివ్ యూ అనాలిసిస్ విచ్ ఈస్ రిక్వైర్డ్. మీరు ఆల్రెడీ ఒక ఇంటర్మీడియట్ పిల్లలు అయితే 2 years, లాంగ్ టర్మ్ పిల్లలు అయితే 3 years చాలా కష్టపడి ఉంటారు. అండ్ లాస్ట్ ఇయర్ కండిషన్లో కూడా లేరు. మీరు లాస్ట్ ఇయర్ అయితే పాపం చాలా ప్యానికింగ్, ఏంటి ఏమవుద్ది అని. అలా కాకుండా ఎగ్జామ్ రాశారు, బాగా రాశారు, మంచి మార్క్స్ వస్తాయి. కట్ ఆఫ్ ఏంటి వీటి మీద ఎక్కువ ఆలోచించకండి, అవన్నీ మీ దగ్గరకు వస్తాయి. అప్పటి వరకు హ్యాపీగా ఉండండి, మీ టైంని ఎంజాయ్ చేయండి, స్ట్రెస్ నుంచి బయటికి రండి, కాస్త రీజువినేట్ అవ్వండి. ఇట్ ఈస్ టైమ్ దట్ యూ నీడ్ టు హీల్ యువర్ సెల్ఫ్. చాలా మెంటల్ ట్రామా లాస్ట్ 1 monthలో మీరు గడుపుంటారు, మీ పేరెంట్స్ గడుపుంటారు. కాస్త మంచి టైం ఇవ్వండి, పేరెంట్స్కి టైం ఇవ్వండి, ఇంట్లో మీకు మీరు టైం ఇచ్చుకోండి. అండ్ ఎనీవేస్ నౌ ఐ కాల్ అపాన్ అవర్ బయాలజీ టీచర్స్. వీ హ్యావ్ కునాల్ సార్, వీ హ్యావ్ రోహిణి మేడం అండ్ వీ హ్యావ్ జ్యోతి మేడం హియర్. అండ్ సార్ వాళ్ళు వస్తున్నారు ఈరోజు పేపర్ డిస్కషన్ గురించి. సార్ ప్లీజ్. Thank you so much and stay tuned. We will give you all the other updates. Next chemistry, తర్వాత physics కూడా solve చేద్దాం మనం. Thank you అండి.
Yeah, so hello students. So I know that you all must be very keen that quickly the answer key should be given. So everything happens in a step by step manner, right? So right now don't worry about cutoff. So first of all let's start subject wise. So first we will cover biology. So overall if we consider biology, so please ma'am, you also come. So in biology if we say, then there is no surprise in biology. If we say biology was typical, it was maybe a little lengthy, and lengthy means what? It was proportion, right? It was proportionate, that you have to give time to bio equally. Paper has taken some time, but those who have prepared well, right, you must have done very good, right? So overall paper was not outside NCERT, it was exactly line by line, question by question, exactly from NCERT, nothing else. So further ma'am for zoology and ma'am will also talk about botany further. So ma'am, why don't you tell how the subject wise the division of different sections.
Ma'am, please. Good evening, my warriors. So I feel you did your best in the biology, right? So we are doing here the analysis for the biology portion. So here ma'am is, Jyoti ma'am is for zoology and me for the botany. Up to the half of the paper for botany I will be here. Then Kunal sir will take over the next of the questions, right? So among the botany, we felt the distribution among the chapters. Like if I'm taking +1 and +2, then we found the +2 portion in botany portion was more as compared to second year portion, right? But I feel, ma'am, it is opposite in zoology.
Yeah, in zoology, majority of the questions are from second year. Second year questions are more comparatively to first year. Yes.
Okay. And analysis, if I do based on the match the column and the statement, multiple statement based,
Yes.
then in botany we found match the column was 9 and the multiple statement type it was 8, and it is surprisingly there is no assertion and reasoning.
Yes.
Right? So see how much you were relaxed in the biology. I felt 360. Many of your students are going to score here, and already we have got the messages. They are very happy with the biology, the LT, what we have put the program, and based on the module book question paper you did your best. Awesome. Hats off for all of you for your hard work, right? And Jyoti ma'am for the zoology.
Yeah, in zoology, majority of the questions we got in statement wise, ma'am. Statement wise is 15 questions, and match the following questions are 7, and remaining almost okay. 2 flowchart questions are there, remaining are easy questions only. So definitely they will score 180 by 180 in zoology, I feel.
Okay, now without any delay, let's start with the paper analysis. On the screen, you will find the question from the code is the 12 here, right? So we are taking code 12 on the screen, fine? So just take out your question paper. If you're eager to find out the answer, I know that answers are confirmed. So let's see, just for a formality also, few question maybe a dilemma you are, let's clarify that. So let's start from 91. The first question is from the zoology.
Yeah, first question is from zoology, and it is very easy question here. Based on elimination mode, you can go here. So Evil quartet biodiversity loss includes. So by removing this air pollution, water pollution and soil pollution, directly you can go for second answer. It is very easy. Next question from botany.
Okay, beta, let's have a look on this question. Okay, so let's see the next question on the 92, where you are having which one of the following is a site for active ribosomal RNA synthesis. First of all, let me to say that already marked answer, don't check that. What we are announcing, that answer should be considered. That is a team overall efforts we did to get the best answer for you, right? Don't look to the answers which are already marked. Fine. I'm reading once again. Question number 92, which one of the following is a site for active ribosomal RNA synthesis, right? So no doubt here, time being the answer has been given, right? Nucleolus, right? But many of the next of the questions what we are going to read, maybe the answer what already marked and what we will say will not match, but right now it is matched here. The first year question, and even that was a direct question from your module books and from the one of the internal paper. We said that, so I feel it will is going to be means in the future also, students those who are following us, it will be useful for you also, those who are listening this all the session. And to our past student, they already get well adapted to the module pattern, and they have written definitely the right option for this question. Okay, ma'am, please take the next.
Yes. Next was
Next is from botany.
Botany
Again.
only.
Okay, so here now list 1 and list 2. This question is based on the chapter cell cycle and cell division. So let's see the G1 phase. If I am taking that one, so you are having here the G1 phase with which we can match it. It is going to be the cell is metabolically active. Cell is metabolically active like throughout the cell division, but the preparating phase, it is going to be more active. But now the hint is that continuous growth but does not replicate its DNA. So obviously the A is matched with the 2. S phase is now very famous for the synthesis of the DNA. So B will go with the 3. Then after that G2 phase, here also protein synthesized while the cell grow continuously, because this is a continuous process, right? And tubulin protein is mainly formed in G2 phase. M phase is actually the main phase, that is actual cell division occur. So it goes with 2, 3, 4, 1. So answer is going to be first option over there. For question number 93, answer should be first option. Fine.
Yeah, next one, this is zoology. In class already I said here, every year they are going to give this productivity, NPP and GPP question. Even this is from our module question also. So this time they have given all definitions at a time as a match the following here. So simply productivity. Productivity means here rate of biomass. This is 3. Next NPP. NPP equals to formula GPP minus respiratory loss. And next GPP definition. GPP means here rate of production of organic matter. Wherever organic matter is there, we have to go for GPP. So this is 4. And next secondary productivity means I said when the option is having the formation of new organic matter, that is the one. So 3142. 3142 means here the answer is 2. Yes, next question. Next question here. Even this is also from zoology only. The question is regarding to which statement is correct. First one is here, the Amazon rainforest. It is being cut and it is a best example for habitat loss. Yes, this is right. Second one is stellar sea cow and passenger pigeon, they become extinct due to over exploitation by the human. Yes, this is also right. Third one is here, the Nile perch. They said in introduction into the East Africa helped in population growth. No, they are not going to help in the population growth. They are going to extend the fishes here. So this option is wrong. Next D, water hyacinth. This is a weed which acts as an invasive species, right? And E1 is when a species becomes extinct, the plant and animal species which are associated with it are also affected, but they have given not affected. So this is wrong. So which of the following are correct means here A, B and D. The answer is 2. Next question.
Okay, next question is from botany portion. Let's check on this. Identify the correct statements about the biomolecules, right? From +1, we are having this biomolecules chapter. Lipids are journey water soluble, which is not possible. It is not water soluble. Then proteins are polypeptides. That's right. That is a long chain of amino acid. Polysaccharides are the long chains of sugars. That's right. Monosaccharides, they are the chain of that. Adenine and guanine are substituted pyrimidines. This is wrong. These are the substituted purines. Almost all enzymes are protein. This is right. Direct statement from your NCERT and very important line. Okay. Now check the question carefully. They are asking identify the correct statement. So correct answer for that means B, C and E. B, C and E means second option will be your answer for this question. Let's check on the next now. How many ATP NADPH molecules are required to make one molecule of glucose through the Kelvin pathway? I feel this is again very easy. There is no calculation. Obviously it is there, but direct this table have been given in your NCERT book. So you can directly opt for your answer. So here will be 18 ATP and 12 NADPH. For how many glucose molecule? One glucose molecule means 6 cycles, and we will get the first option as an answer on question number 97. Okay, now coming to the next question number 98. In question number 98, you will find this is from your +2 course from the biotechnology. This question is based on restriction endonucleases, and see what they are asking, which is not true. So if you have read your question very carefully, not true, correct, so it will really help you to get you your right answer. Let's check on this now. They are called as what thing? The molecular seizures. This is right, fine. Then these are the enzyme responsible for restricting the growth of bacteriophages in E. coli, right? Because they are, you can say, break the DNA of viruses to restrict its growth inside the prokaryote or like a bacteria. Then they cut the DNA only at the center of the palindromic sequence, not because we are having even slightly away from the center, making stagger ends, sticky ends like that. So it is not correct. They remove the nucleotides only from the end of the DNA fragments, not at all. It is from the center or within the DNA also, not exactly on center, within the DNA and even on the edges also. But the endonuclease is within. Nucleases can be within or extreme, but endonuclease is within, not on the ends of it. Then they recognize specific palindromic base pair sequence. This is right. But again, be careful, not true. So which is not true? C and D. So your answer comes out to be third option for this. Okay. Let's check on the next question. This is from zoology now. Okay.
Yes. Yeah. So this is direct question from NCERT. So here simply logic here, by taking detritus, it is 4. So B is 4. Only one option is there having B is 4. So direct question. This is direct NCERT question. Even we have studied many times here. So decomposition. Decomposition means breaking down of organic matter into inorganic matter. This is 3. And mineralization means release of inorganic nutrients by the microbes. So this is 2. And humification means formation of humus. Formation of humus is known as humification. So the answer is 3. Yes.
Okay, next question is from the botany again. Very easy question. In which one of the following the ovule are not enclosed by an ovary wall, right? Remain, and means ovules remain exposed. Many times we have discussed in the class also, even in module book directly given, even in internal paper many times we have asked this question. So here Slegionella is a pteridophyte, not having the seeds. Funaria, not again, it is not a spermatophyte again. Pinus, yes, it is a gymnosperm, comes under spermatophyte seed bearing, but it is not enclosed with the fruit. Right, Wolffia is belonging to angiosperm, their seeds are enclosed inside the fruit. So, but they are demanding the ovule which are not enclosed in an ovary wall and remain exposed. Answer will be Pinus, being it is one of the example of gymnosperms. Okay. Now coming to the next, that is from your chapter morphology. There again, many times we have discussed, LD students have responded very nicely on this question many times. Right, let's see. What will be the answer for this setup of the, you can say placentation, and with their example. See here, the marginal placentation we can match with the pea here, okay. Axile placentation we can match with the lemon. Then parietal placentation we can match it with the mustard. And basal placentation we can match with the marigold Asteraceae family, okay. So 2, 4, 1, 3. Answer comes out to be third option for question number 101. Let's take on the next question now. Again, this question is from botany part. It belongs mainly to the morphology, and even in the case of plant growth and development, you will find a little intro about this also. In angiosperms, root hair arise from which one of the following regions of the root? They are asking about root hairs, beta. The root hairs are the extension of epidermal cells, and you will find in the region of the maturation. The answer comes out to be fourth option. 102 question, answer will be fourth option. Coming to the next question, they are asking which of the following is not a characteristic of plant cells in the phase of elongation. Increase vacuolization, that's right, vacuolation started in the elongation, that's why water enter, expand, and it is getting elongated. Large conspicuous nuclei, it is not a feature of elongation. This is for the meristematic region, which is having conspicuous, very clear nuclei over there, and cell enlargement will happen again in elongation. New cell wall deposition will start in the elongation. So means which is not belonging to elongation is a second option, large conspicuous nuclei, for question number 103. Let's take on the next question. Here they are asking which of the following statements are correct with reference to a transcription unit. This is one of the very important chapter, and that is statement based question. If you have read every statement very carefully, I thought you have got your answer. Which of the following statements are correct with reference to the transcription unit, beta? So for this, if I am taking that, okay, so here we are taking for, okay, again I am taking which of the following statements are correct with reference to the transcription unit. Okay, if you remember, this is a transcription unit. Here we are having 3' to 5', here 5' to 3'. We are taking here the promoter, and here we are taking the terminator. This is the template stand, this is the coding stand for the structural gene. Fine, beta. Now let's take on the statements. In transcription unit in DNA is defined primarily by 3 regions: promoter, structural gene, and terminator. This is a right statement. Then after this, promoter is said to be located towards the 5' end of the structural gene, because all the references we are taking based on the coding stand. Direct line in your NCERT. Then the promoter is a DNA sequence that provide the binding site to whom? That is to the RNA polymerase. This is also the right statement. Okay, beta. Then after that, the promoter defines the template and coding stand. That's also right. If you interchange its position, that even is going to interchange with the template stand and coding stand. The promoter defines the template that just now we have discussed. Then E is what? The terminator is located towards the 3' end of the coding stand, and it defines the end of the process of transcription. That's right. This is upstream, this is a downstream. So obviously this is an end of the transcription. So first option is your answer here, where A, B, C, D and E, all statements are correct for question number 104. Okay, then after that, coming on the next question, 105. Again, this is from your botany portion, biomolecules, that is from the +1 portion. Alpha helix is found in which level of the protein structure? Very commonly you had found in the internal paper. LT student again respond brilliantly on this also. Here 105, here alpha helix is at which? Secondary structure, which is making the peptide bond and balanced by the hydrogen bonds. So answer should be fourth option for 105. Coming to the next question, 106. In 106, which of the following statements are correct regarding amino acid? Again, biomolecules. See how much important that chapter is. The way we have discussed, and the question came in internal paper, linked to the molecular basis of inheritance also, right? So here, which of the following statements are correct, beta? We have to check it regarding what thing? Amino acid. They are substituted methane. This is right statement. Their hydrogens are replaced by amino group, COH group and the variable group. 1H will remain as such. This is right. Serine is an aromatic amino acid. Wrong, because this is an alcoholic amino acid, right? Valine is a neutral amino acid. This is right. Then lysine is an acidic amino acid. This is wrong. This is a basic amino acid. If you have confusion with the lysine, one of the internal paper, again histone, where they have given lysine arginine mentioned there as an basic amino acid. So if any doubt raised on this, might be you have cleared from there also, right? So here they are asking which are the correct. So which are correct here? A and going to be the A and C. So answer comes out to be third option for this, right? For question number 106, A and C are correct, means third option is correct here. Okay. Now coming to the next question number 107. Here beta, the main function of bulliform cell, very easy question, means in the grasses they are talking about monocot leaf, right? Bulliform are on the adaxial surface. So here to make the leaf impermeable to fungal spores, not at all. Its function is not for protection like this. To perform photosynthesis, no, this is going to be empty large. They are going to take water to open the leaf, and if it is going to be flaccid, then invert curling will happen. So means to minimize the water loss during the water stress. It is playing its maximum role over there. Then to transport water, not at all. This is not for conduction of water. So question number 107, answer should be third option. Okay, beta. Now coming to the next question, 108. In question number 108, again this is based on photosynthesis first year. I already mentioned first year questions were more here. So let's take on this question now. Here we are having find the incorrect. Again they are asking incorrect statement about photosynthesis from the following. Water splitting complex is associated with PS1, not at all, because this is associated with PS2. Okay beta. Then after that, C4 plants use the C3 pathway of carbon dioxide fixation as a main biosynthetic pathway. That's right, all plants, they have to show the Calvin cycle, which is the main site for the sugar formation. In C4 plant, photorespiration does not occur. This is also right, being the ribisco is protected in the bundle sheath cell where it is having Casparian strip, right, cut off from the oxygen. C3 plant exhibit the trans anatomy. No, that was the C4 plant. Those who are exhibiting trans anatomy. ATP synthesis in chloroplast occur through chemiosmosis. This is right. But we have to choose which are incorrect statement. So which are incorrect here beta? A and D. Answer comes out to be second option for question number 108. Okay, after this, next question is also based on the botany. Match the column. Here mark the list 1 with the 2. This is respect to anatomy part, transverse section. Now here conjunctive tissue is where? Conjunctive tissue is present between the xylem and phloem. So A you can match with the 3 here. Okay. And then Casparian strip. It is in the root transverse section again. In the endodermal cell, this is a suberin thickening. It goes with the 4 here. Then after this, subsidiary cells. Subsidiary cells are the cells which you will find, like if I am taking for specialized cells in the vicinity of the guard cells. It is there. Subsidiary cell is near. Vicinity is very close to the guard cell. It can be present or absent also. So it is going to match with the first. Then starch sheath beta. Starch sheath is going to carry the starch inside it. Endodermal cell rich in starch in the dicot stem. Okay. So it is going to match with the 2. 3, 4, 1, 2. So answer going to be the third option for 109. Okay. Now coming to the next question. 110, again match the column, but it is based on your +2 syllabus. So genetically modified organism, that is the BT cotton, right? Then thermostable DNA polymerase, which we are using in PCR, that we are getting from Thermus aquaticus. TI plasmid, we are getting from Agrobacterium tumefaciens. Their T DNA is causing the tumor in the dicot plants. PBR 322. That primarily plasmid we have taken from the Escherichia coli. So 2, 3, 1, 4, answer goes to with the third option. 110, third option will be your answer. Okay, now next is again from your +1, plant growth and development. 111, that heterophilous development in response to environment is an example of which of the following phenomena? So answer is going to be the plasticity. This is not only with the environment also, it can be the developmental stages also. Means any of that is given, but this is the right thing, we are matching with the plasticity. Very easy question. They are not asking any example and all that, directly they are asking the process name. So 111, answer should be the fourth option. After this, coming to the next 112. In that case, in racemose inflorescence, again better no family have been asked, no example, directly features have been put forward for racemose and we have to select the right answer for this. The main axis terminate into a flower, this is for cymose. Then the growth is limited, again for cymose. Then flowers are born in an acropetal succession, this is your answer, right? flowers are solitary. No, this is going to be for some cymos, when, or you can say category solitary separate, then we are having cymos and racemose, or sometimes solitary we are taking under the cymos, but definitely this is not for the racemose. 112 answer goes with the third option. Then after this, next question, that is from your zoology part.
Yeah, this is directly from NCRT, even our module question also. So, which of the following disorder is caused by means of substitution? Means it is about point mutation. Point mutation example is here, sickle cell anemia. The answer is 3. Yes, next question.
Okay, beta. Next question is again from botany part. Here they are asking for matching, and botany second year beta. Incomplete dominance beta. It is going to be matched with the inheritance of the flower color in anterinum. It is going to be matched with 2. Co-dominance with 4 ABO blood group 4. Then pleiotropy is for the you can go with the phenylketonuria 3. Then after that, polygenic inheritance, human skin color. So it goes with the 2431. 2431 means first option, 114. Okay, then after this, arrange the following in a correct developmental sequence related in microsporogenesis, where we have to discuss the process in the anther when it is happening in the male. So first they are saying microspore tetrad. I am keeping here for a while. After this, the sporogenous tissue. So obviously that will be coming first. Pollen grain will be at the last. Then pollen mother cell is after this. So BDAC sequence come, answer should be third option for 115 question. Okay, after this beta, we are having arrange the following for DNA fingerprinting. This is from your zoology part.
Yeah, this is from DNA fingerprinting. Direct question here. Even the points are given in the textbook. We have written this question during our weekend exam as well as GT also. So first one is here, isolation. Without isolation, we cannot start the DNA fingerprinting. So the first step is isolation of DNA from the crime scene or either from suspected person. After isolation, second one is here, we have to cleave it by using a restriction endonucleases, then separation. Second point is separation. After separation here, transferring. Transferring means here, this is a kind of blotting technique here. After blotting is done, we have to hybridize. Hybridize by using VNTR probe, and finally detection by means of autoradiography. Then the answer is here 1. Yes. Next question. Yeah. So this is from in situ question, in situ hybrid, in situ 1, and this question is there in our brackets here. So exploring the molecular, genetic and species level diversity means here, it defines about bioprospecting. Answer is 4. Next question. Next. This is about sex determination in honeybee, and they are asking about true. It means correct statements we have to find out. First one is, an offspring formed by the union of sperm and egg, they are developing either into male or, that is, they are developing into female, that is either queen or worker. Yes, A statement is right. Unfertilized egg develops into male by means of parthenogenesis. Third one, a male has half the number because male is haploid and female is diploid, means it is half. So C is also right. D1 is males produce the sperms by not meiosis, it is by means of mitosis. E is their honeybee sex determination is by means of haplodiploid method only. Okay, even this is also right. Only D is wrong. Wherever the option is D, simply you can eliminate. Then you can find direct answer 4. Yes, next question.
Okay, next question again from botany +2, your biotechnology beta, very favorite question of NTA, PYQ even covered that. Identify the correct sequence of the steps in each cycle of polymerase chain reaction. So denaturation happens first, then annealing of the primers happen, then extension happen. So answer should be first option. This is the correct sequence of the event happens during the PCR technique. Okay, then 120 again is from botany beta. Which of the following statements are correct? Find which of the following statements are correct with respect to DNA separation, isolation and visualization. So here the cutting of the DNA is done by molecular scissors. That's right. That is the restriction enzyme. Then the DNA fragments separate accordingly to their size in agarose gel upon electrophoresis. Smaller are moving faster and the bigger one are still towards the well. This is right. The separated DNA fragments can be seen without staining. This is wrong. We have to stain it by ethidium bromide when exposed to the UV radiation. This is wrong, beta. Then after that, the separated DNA fragment when stained with ethidium bromide can be seen in a visible light. This is wrong. It is not due to visible light. This is due to UV light. So they are asking for the correct. Which one are correct? A and B. Answer should be fourth option. Now rest of the botany question will be handled by Kunal sir.
So, hello students. So, the paper discussion is going on and I can see that many of you are taking name of your favorite teachers and asking for cutoff, that you are asking repeatedly. But before moving ahead, all of you those who are connected, first tell me whether your paper was easy, you find the paper easy, moderate or tough, right? Just write there that was it easy, moderate or tough, and then side by side we are discussing the paper. So till 120 it is done. See, this is R.H. Whittaker, right? So this is again direct NCERT question and the same question was asked in the grand test also. Exactly same question, right? So as you can see here, the correct answer here, you can clearly see presence of flagella. It is not a criteria, right? Presence of flagella is not a criteria, rest all are the criteria, right? So here the correct answer will be A, B, D and E. Rest all are correct. Then next question. Again, this is again a very, very important question. I think the ploidity question was asked at least 5 to 6 times in the grand test paper. Here the triploid, so 3N. So 3N will be primary endosperm cell. Primary endosperm cell. So many of you are telling paper is lengthy, easy but lengthy. Yes, paper was easy. Biology, why you think that bio you can cover in 30 minutes? Bio will take 1 hour, right? So it is not unusual, but most importantly it is easy. Yes, next question. Now, this one, it is asking about DNA helix. So which of the following statement is correct with reference to packaging of DNA helix? Again, direct NCERT question. Histone is organized to form a unit of 8 molecules. Histone is an octamer, correct? Histone is negatively charged, right? Histone is negatively charged, wrong. Histone is rich in basic amino acid residue. Histone is rich in basic amino acid residue and the positively charged DNA, right? The positively charged DNA is wrapped around the histone octamer. So here the correct answer will be for question number 123. The correct answer is 2. Here which A, C and E is correct, rest are wrong. Then question number 24 is about zoology.
Yeah, it is from ecology. They're asking about in situ. In situ conservation here. Out of 4 options, only first one is in situ and remaining 3 are ex situ. So direct question here.
Yeah.
Next question.
So again, straightforward question, Z gene, right? So in lack of operon, the Z gene code for beta galactosidase. A very, very easy question. Next is the match the following question. As you can see again, 2,4-D. 2,4-D is a herbicide. Even if you match one, you will get the answer, right? So here A will go with T, then B will go with 1. So correct answer here is 3. Third is the correct answer here. Then next one, arrange the following steps of somatic hybridization in correct sequence. So for somatic hybridization, you need cell from 2 different variety of plants. So that is the first step. Then you will digest the cell wall. After that, you will isolate the naked protoplasm, then fusion, and finally growing. So correct answer will be 2 here. Next. Now, if you remember, this is also asked in grand test and it was also asked in weekend test. Same equation related question was asked twice. So here if you see the respiratory RQ is how much? It is less than 1 and it is between 0.5, 0.9, actually exactly it is 0.7. So correct answer will be 2. Then next.
So this is about extinction. How the 5 episodes of previous extinction was different from present 6th extinction. Means answer is simple answer. The present species extinction is 100 to 1000 times faster than the previous one. Direct question here. Next one.
Next one again match the following. So here you can clearly match trypsin. It is a enzyme, right? Trypsin is an enzyme. B, morphine will be alkaloid. Then concanavalin, C will match with 2. And then collagen is the intercellular ground substance. So correct answer will be first. Next, binomial nomenclature, very simple and easy question here again. So here if we see both the words in binomial biological name were handwritten, separately underlined, correct. A specific epithet starts with small letter, correct. The first word is represented, this is wrong, first word is genus, second word is species. So third is the correct answer here. Then next, the enzyme required for carboxylation in Calvin cycle. So it is Rubisco, very simple, straightforward. Next one. So again here, simple question, as I have said, don't worry about families. Family will always, if there is one question or 2 question, it will be the simplest question. And I said the one which is there in NCERT, that is more important. So that you can clearly see that Solanaceae family. So here the correct answer will be, here the first one is the correct answer. You can clearly see this is the floral formula. If you remember, I have also told in class that floral formula, floral diagram, example, these 3 things you should focus. Question is most likely from here and you can see this is the question. Next one. Again here, which of the following type of pollination bring genetically different type of pollen grain? So it will be xenogamy, right? That is the correct answer. Yes, next. Again, very simple question. Very simple question from match the following. So again, this, if you see, it is a match the following and it is also very simple. If we say it is the location where these process happen. So glycolysis, where does it happen? It happen in the cytoplasm. Now, if you just check here, A3, only 1. So even if you know 1, you can tick the correct answer. So second will be the right answer. We all know all this. So this is the right answer here. Next. Insertion of foreign DNA at BAM-H1 site. Again, we have seen in class, we have discussed that PBR 322 is very important. And at least 10 to 12 times during grand test and weekend test, question is asked from the structure of PBR 322. So very simple question. PBR 322 BAM-H1. So we know that BAM-H1 is present at which site? So it is present at tetracycline site. So tetracycline will be lost. So that third will be the right answer here. Then
Next one is again sickle cell anemia question here. They are asking mutant codon. So which of the following is a mutant codon of beta globin means here? The answer is simply GUG. Because normal codon is GAG. GAG is going to mutate into GUG. So the answer is here 2. Next one, reproductive health. They are asking about correct statements. Correct statement about GIFT, gamete intrafallopian transfer. Here in the first option they said transferred to uterus, so this is wrong. Second one is transfer of ovum collected from donor into fallopian tube who cannot produce ovum. So this is direct line from NCERT, this is right. And third one is this is about embryo, not embryo, we are going to insert gamete. So embryo wrong, here also embryo wrong. So the answer is here 2. Next question is here, example of sexual deceit. Answer is, one and only answer is Mediterranean orchid as well as bumblebee. Next one, this is evolution one. Simply we can go through our mnemonic Drahen here. Drahen means Dryopithecus. Here they have given Ramapithecus in between, so this is wrong. Here also Ramapithecus has given after Homo sapien, wrong. Again this is also wrong. Here Drahen, first one is Dryopithecus, next Ramapithecus, Australopithecus not given, Habilis, Erectus, Neanderthal and Homo sapien. The answer is here 2. Next, this is from reproductive health here, human reproduction chapter, gestation development. The first one is here, the fetus movement starts. When does the fetus movement will start? It will start at 5th month. 5th month means here 20 weeks. So this is 2. So A should be 2. Next, fetus develops limbs and digits. Limbs and digits means it is 8 weeks of pregnancy or 2 months here. 2 months. So this is 3. B should be 3. B should be 3 means here the answer is 4. Direct and easy question. Next one. Some fish-like animals, upon investigation, the following characters. Endoskeleton is made up of cartilage and they are ectoparasites. Ectoparasites means here they are cyclostomes here. And they are having a circular and sucking mouth. So all these features are explaining about cyclostomes. Exocetus is not a cyclostome. So the answer is here Petromyzon. Next, spermatogonia. This is about spermatogenesis. We have to select about correct statements. Spermatogonia always undergo. So they have given this keyword here. So this is wrong. Always undergo meiotic cell division, wrong. First it has to multiply by mitosis and then meiosis. Second one is primary divides mitotically, not mitotically. It is meiosis 1 here. So the second one is wrong. Third one, secondary spermatocytes undergo second meiotic division, produce haploid spermatids, right? D is spermatids produce spermatozoa through not mitosis, it is by means of differentiation process. Next, E1 is spermatids transfer into spermatozoa by spermiogenesis. Yes, this is also right. So correct statements means here C and E. C and E only, first option.
Yeah. So again, I think this is the only numerical based question in botany. So as you can see here, what is the probability of having a children with O blood group? Both are heterozygous. So heterozygous means AO and BO, right? This one is if they are heterozygous, so chance of getting O here from here is 1/2, from here also 1/2. So it will be 1/4, so 25%. Simple question. Next.
Yes. Next, about this RAS mechanism, they are asking order. First one here, fall in GFR. Fall in GFR, it will stimulate the JG cells to secrete renin. So then the renin converts angiotensin into angiotensin 1 and later on into 2. And this angiotensin 2 acts as a powerful vasoconstriction. So this is 3. After that, aldosterone will be produced. This aldosterone acts on lateral tubules and it involves in reabsorption. Finally, it increases the blood pressure and GFR. Then the answer is 4. Next question, this is from respiration chapter. Tidal volume, actually we will go easy way. So tidal volume is 500, means D should be 2. D should be 2 here. Next one, RV. RV is here 1100 to 1200. So B should be 4. Then the answer is 3. Yes. Next one, reproductive health. Progesterone. Progesterone means it is a type of IUD, hormonal IUD, and multi-load is copper releasing IUD. Diaphragm is a barrier, whereas Saheli is a oral contraceptive. Then the answer is A is 3, B is 4. Then first option.
So again, a very simple question in botany. Non-membrane bound cell organelle found in both prokaryote and eukaryote, simply ribosome. It is common both in prokaryote and eukaryote. Next.
This is also direct NCERT question. Even we have written in GT as well as weekend. One and only pyramid which is generally inverted is pyramid of biomass in C. Next, question is about flightless bird. Flightless bird. Flightless bird means here even Shrutio also will come as well as actinoditis. But they are asking forelimbs are modified into paddle-like structure which is swimming. Swimming means the answer is penguin. It is an aquatic and flightless bird.
So here this is again a match the following. So here if you see lipase. So lipase is used in detergent formulation, right? So lipase is used in detergent formulation. Even if you just match one, you will get your answer, right? Then statin. So if you see this, so overall you can say first one is the right answer. If you match all of them, this first is the right answer. Next. It is again here a very simple question. So if we see here, the endomembrane system include Golgi complex, ER and mitochondria. Is mitochondria part of endomembrane system? No. Rough endoplasmic reticulum bear ribosome on its surface. Correct. Both mitochondria and plastid have circular DNA. Correct. A network of microtubule, microfilament and intermediate filament present is called cytoskeleton. Correct. Mitochondria is single membrane bound structure. Wrong. So B, C, D is correct here.
Next one, animal kingdom question. They are asking about bony fishes. Which of the following set of fishes are belonging to the bony fishes? Simply devilfish and cuttlefish, this is wrong. Here starfish wrong, and here dogfish wrong. So then the answer is here third one. Next, grasshopper. Grasshopper belongs to XX and X0. So this is female and this is male. It means male will have less number of chromosomes compared to the female. Then the answer is possesses 24. 24 means here female. Here 23 and 24 means 23 is male and 24 is female. Then the answer is here males and females respectively question here. So fourth option. Next one, this is the calculation part here. They are asking number of WBCs are 8,000. And what is about eosinophils? Eosinophils percentage are 2 to 3%. 2 to 3% means here 8,000 is 100%, means 1% is equals to 80. Here 2 means here it will be 160 to 240. Whereas lymphocytes are 20 to 25%. 25 to 25% means here 10% will be 800. Then 1600. So then answer is here first one.
Yeah, so again here this is a very important question. We have discussed it in class that, and I have mentioned that these genes may be a little confusing, right? So some of you might have got confused with the options here, right? So as you can see here, to cotton bollworm and corn borers, right? So they both have different genes present. So here the correct answer will be third for cotton bollworm, cry1AC and cry1AB for corn borer. So third is the right answer here.
Next, match the following question here. By taking morphine here, it is a sedative and painkiller. So B should be here 3. Next, nicotine. Nicotine is going to stimulate the adrenal gland. So A is 2, 2, 3. Next, heroin. Heroin is actually depressant. C should be 4. Then the answer is 2. Next, again one more match the following question. Tetany means here it is a rapid spasm due to low calcium levels. So this is 3. Arthritis 1. Myasthenia gravis here. It is an autoimmune disorder affecting the neuromuscular junction, and muscular dystrophy means it's a kind of genetic disorder where a progressive degeneration will take place. So 3124. First option. Next, in which animal do haploid cells undergoes mitosis to produce gametes? Means that is only male honeybee. Next, this is flowchart question here. Even I said a sequence. First one is here pulmonary ventilation. Means first one should be C. And last one should be cellular respiration. D. Next, in between transport you have to take. So, in between E should be there. The answer is here 4th. Next, this is diagrammatic question here. They are asking layers around the female gamete from outer to inner. Outer to inner means first one is corona radiata. After this, zona pellucida. Next, some space will be there. After that, plasma membrane. Then answer is CABD. Answer is CABD here. Next, antitrypsin. Direct question. Antitrypsin we are using for the treatment of emphysema.
Yeah. So here you have to find the correct statement. So if we see membrane of the human RBC has approximately 52% protein, right? Correct. Major phospholipids are arranged in bilayer, again correct. Extension of plasma membrane into the cell to form mesosomes. Now, if you haven't read the question very carefully, right, then you might mark it correct. See, it is for eukaryotic cell. Mesosomes are found in prokaryotes. So this is also a wrong statement. Then the tails toward the inner part of the lipid are hydrophobic and thus protect from aqueous medium. So this is also correct. Glycocalyx is present on the outer surface. Glycocalyx, again eukaryotic. So this is also wrong. So here the correct answer will be A, B and D.
Next one is about frog. They are asking male frogs can be distinguished from the female frogs. First option, bulging eyes is both. Ocal sacs are only for male and webbed feet is for both male and female. This pad is for male and green color, this is also for both. But they are asking male, only male here. So the answer is here B and D only, first option. Next question. This is direct question. Logistic population growth here, first one. Next again about frog, correct statement they are asking. Hepatic portal system is a venous connection between liver and intestine, NCRT line, right? There are 12 pairs, not 12 pairs, it is 10 pairs here, wrong. Third one, the uterus and oviducts open separately in the female, correct? Hindbrain consisting of cerebellum, medulla, correct, but optic lobes are wrong. It is a part of midbrain here. And sinus venosus joins to right atrium, right. So the correct statements are here A, C and E. Second option. Next one about RH grouping, incorrect options. First one is here, it is observed having fetus with negative blood and mother with positive. Reverse they have given. Wrong. RH antigen is absorbed on RBC in majority. Yes, 80% is RH positive only. Before blood transfusion, RH grouping also should be matched, right? RH incompatibility is observed when pregnant mother is negative and fetus is positive, right? This fetalis can be avoided by administrating anti-RH antibodies to the mother immediately after keyword correct, but yes, second they have given. It is not second, it is first. So this option is wrong, but we have to select incorrect. Incorrect means here A and E. First option. Next one, regarding to human endoskeleton, correct options here. Human skull is not mono, it is dicondylic. The joint between any two adjoining vertebrates means it is intervertebral disc, okay, right? In human beings, the number of cervical are 7. All ribs except last pairs, no, all are bicephalic. All 12 pairs are bicephalic only. The occipital bone of skull articulated with atlas, right? So the correct statements are here B, C and E. Next one, match the following. Cortisol. Cortisol produces anti-inflammatory reaction. Aldosterone stimulates reabsorption, and this is an enzyme from GIT. It stimulates the pancreatic enzymes. This is 4. Progesterone stimulates the formation of alveoli. This is the one. 2, 3, 4, 1. Second option. Next one, again order, proper order. First one is, this is from diagram question here. First thing is, female mosquito has to bite. Then it is going to inject sporozoites. These sporozoites are reaching the liver through the blood. After reaching the liver, those parasites undergoes reproduction asexually in the liver. Then it will reach us to RBC, again undergoes asexually, 4, and finally in RBC they will change us into gametocytes. So this is 5. Then EDB. So second option. Next, incorrect. Digestive system in platyhelminthes is incomplete, correct? Bilateral is not in adult echinoderms. It is actually radial symmetry. Penta, radial symmetry here. Pseudocoelom is possessed in Aschelminthes, right? Notochord is persistent throughout the life in the class Chondrictitis. Actually, this is a NCERT line. Next, members of class Reptilia. No, they cannot maintain because reptiles are poikilotherms here. Which are incorrect means here B and E. B and E are incorrect. This is a very easy question here. Where are these receptors are present? These receptors are present always on postsynaptic membrane only. Next again, sequence question here. Correct statements. A motor neuron carries a signal sent by CNS, right? Sarcolemma also correct. Neural signal generates an action potential which causes release of calcium into sarcoplasm, right? Increase of calcium not inactivates, it activates. Actin binds to myosin to form a cross bridge, right? Shortening of sarcomere takes place by pulling actin towards the center. So all are correct except C. Then the answer is 1. Next, one more easy question. They are asking about not an example of convergent here. So the first one is convergent. Third one is also convergent. Fourth one is also convergent. Only the second one is about homologous organs are divergent here. So this is divergent. The answer is here 2. Next, JG apparatus. I said JGA. A means here afferent arteriole we have to take. So DCT plus afferent arteriole is equals to JGA.
Yeah. So again, one very super easy question in botany. So direct NCERT question, and this you all must have practiced several times. So this is a question where you have to identify which enzyme is catalyzing this reaction. So very simple. when a double bond is formed, then the name of the enzyme is lyases. So second will be the right answer here. Next, 177, zoology.
Again, match the following here. Molluscans. Molluscans are having a ctenidia, feathery-like gills here. So it is brachial respiration. Reptiles. Reptiles are terrestrial animals. They are having pulmonary respiration only. Adult amphibians means here they can respire either through lungs or even skin also. And amoeba means here by means of body, that means plasma membrane only. So this is 3. 2143.
Again, this is a question from botany, straightforward, simple question. Swiss cheese production, which microorganism is helpful? So here Propione shermani, this is responsible for formation of Swiss cheese due to production of large amount of CO2. So correct answer here will be 4th.
Again from evolution. 65 Maya means here it is where the dinosaurs disappeared. 2. 500 million years ago means Jales fishes. 1. 350 million years ago. 350 million years ago means here it is sorry. Yeah. 65 million years ago means where the dinosaurs disappeared to, and next 500 million years ago means invertebrates were formed and were active, and about 350 million years ago means Jales fishes, and about 320 means here seaweeds. 3. So 2413. It is first option. And the last question is here about population interactions. In both parasitism and commensalism, only one species benefits. No, wrong. Both species benefits in mutualism, right? Both species benefit in commensalism, wrong. In parasitism, one is benefited, another one is harmed, right? In amensalism, one species get harmed and other one is unaffected, right? So, which of the following are correct? Correct means here B, D and E.
So that was the paper discussion of an answer key of biology. So as you can see, it is directly from NCERT. Now all of you just in the chat write your marks, how much you are getting in biology. By the time chemistry sir take overs, you all write quickly what is your marks in biology. Now I will like to call the chemistry faculties to discuss the answer key of chemistry. But all of you write your marks in biology, how much you are getting out of 360.
$320, $345, $345, $355. That's very, very good. So I hope you have attempted your chemistry paper good as well. So the paper level as compared to last year, I would say was little on the easier side, but a little more difficult as compared to 2024 paper. What do you think? How was your paper? So we'll start with the discussion of chemistry. My name is Anirudh, and let us continue with our chemistry paper now. You can tell me in the chat box how was your paper today.
Very easy. Good. That's good. All right, guys. So we'll be starting with our question number 1 here. This is going to be code 12, I guess. So you can all check your answers. Question number 46, which is a conversion type question. How to go from cumene to phenol, which is aerial oxidation followed by hydrolysis. So A matches with 2, there are different options. Then there is acetic acid to ethanol, which is reduction. You can do its esterification and then you can reduce it using your hydrogen palladium, which is the more efficient way to do as written in the NCERT. LiLH4 is a little expensive reagent. B matches with 3. And then we have propanol going into propane 2-ol. So first we have to remove the H2O and then we have to add the water by Markovnikov rule. C matches with 4, which is going to be fourth option. Right. Moving ahead, question number 47. Product Z they are asking. First thing they are doing free radical halogenation, giving you chloroethane. Then reaction with ammonia. Ammonolysis, if nothing is mentioned, do it one time. It gives you ethanamine. And then ethanamine, you are trying to do diazotization. We know that diazonium salts on your aliphatic compounds are unstable. So they will easily hydrolyze to give alcohols. So ethanol would be the answer here, which is second option. Ethanol. Moving ahead, question number 48. It's a nice question, easy question. There is a solid which is dissociating into aqueous, so it's a sparingly soluble salt. It's a AB type sparingly soluble salt, so solubility will be S square. S square will be equal to KSP. The KSP will be S square, 4 into 10 power minus 10. So S comes out to be 2 into 10 power minus 5. As it will dissociate into S mole per liter BiO positive and S mole per liter OH negative. So this is concentration of hydroxide. So we can say from here POH is going to be negative log of this number, which comes out to be 4.70. This is POH. Okay, so don't do a mistake of choosing this option. This is POH, but they are asking pH, which will be 14 minus 4.7, which will be 9.3. Okay, I hope you have not done a silly mistake here by choosing 4.7. pH is 9.3 here. Right. Moving ahead. Another question from your module, and it's a very basic question. It's, I think, a PYQ as well, a very similar question. How carbon dioxide is reduced with coke is it gives you 2 moles of CO gas. This is gas, this is solid, this is gas. If I start with 1 liter of this is 0, this is 0 initially. I mean, this is anyways excess, so excess reagent will not look at. It is passed over red hot carbon. Volume of the gaseous mixture after the reaction becomes 1.4. The composition of gaseous mixture at STP is how much? Means they did the reaction. Okay, this is not given, so we don't know how much this amount is. But if the reaction goes to completion, in that case, 1 liter should have converted into 2 liter. But I didn't get 2 liter, that means the reaction is not complete. So we can just say, let's say reaction, initial, change and final. Let's say it breaks down X liter. So this will form 2X liter. So final composition will be 1 minus X liter will be CO2 and 2X liter will be CO. This total mixture is given 1.4 liter, which is 1 plus X, 1 minus X plus 2X. From here, X comes out to be 0.4 liter, meaning CO at the final stage is 2X, which is 2 times 0.4. This is 0.8 liter. And this is 1 minus 0.4, 0.6 liter. So 0.6 liter CO2 is remaining and 0.8 liter CO is remaining. Third option is correct. Okay, right. Moving ahead. Again, a very simple question. 2P, 404S, 535F and 3D. You can easily match from here. Moving to the next one. Yes, in the presence of anhydrous AlCl3, which is going to be a electrophilic substitution reaction, will give you hexachlorobenzene. All the hydrogens are replaced by Cl. So there are 6 chlorines in this. In the presence of UV light, it will give you addition reaction to give you benzene hexachloride, BHC, benzene hexachloride. This one also is going to have 6 chlorine only. So we can say both have 6 and 6. Second option is correct. Okay. Again, a very simple NCERT based question. Most of the questions are NCERT based. This one. With PCl5, we know 1:1 reaction takes place. 1 Cl goes to your alcohol. 1 Cl goes to hydrogen, HCl, and then oxygen comes with the PCl3. We get POCl3. X is POCl3. So X is POCl3, X is POCl3. After doing alcoholic KOH, you get a propene. And after propene, you are doing anti-Markovnikov addition of HBr. Anti-Markovnikov will give you a 1-bromopropane, which is going to be fourth option. Which will be fourth option. Okay, easy question. Moving ahead. Right. Question coming from D block chapter. The uses of some D block compounds as catalyst. Iron is used for the preparation of ammonia, Haber's process. B2O5 contact process. Okay, contact process. PDCl2 Wacker process, and nickel complex to get the cyclo-octatetraene from the 4 moles of ethine. Right? So it's again a very simple question. You can see the matching over here. B170. Rahul, 170 means botany 170 you got. That's great. Okay. Moving to the next question about ClF3. ClF3, as all of us know, it is going to be a T-shaped molecule with 2 lone pairs on the equatorial position so that the repulsions are minimum. So in this case, we get a T-shaped geometry. You can call it T-shaped or T-shaped geometry. Geometry could be molecular geometry here. So anyways, there is no confusing option. If there was a confusing option like trigonal bipyramidal, then you could have gotten confused, but there is no such option. Trigonal pyramidal is anyways not correct. It has T-shaped geometry is correct with 2 lone pairs on the chlorine. Trigonal planar again not correct. And T-shape with 3 lone pair on the chlorine is also not correct. So we can just say the second option. It's an easy question again, coming directly from the NCERT line to line. Okay. Yes. Coming to the next one. This is one question where many students have done a small mistake. Okay. Generally, here saying EMF of the half cell doesn't have a meaning. They want to say find the potential of half cell. Potential. If the question was asking you reduction potential or electrode potential, then you should have written a reduction reaction. But here, as you see, the cell is mentioned where H2 is converting into H+. That means it is going to be an oxidation potential that they are asking. Okay, not the EMF. Obviously, half cell can't have an EMF. So oxidation potential you need to think of. So write the oxidation reaction. H2 goes to 2H plus aqueous, losing 2 electrons. Correct. And here also you can see SOP is given. So maybe they were trying to give you a hint that talk about oxidation only. Although SRP, SOP both are 0 for hydrogen as we know. So we'll write the Nernst equation, which is E equal to E naught minus 0.06 almost I can say because options are very far by N, which is 2 log concentration of H plus ka square, which is 2 into 10 power minus 2 square, which will be 4 into 10 power minus 4 by pressure of hydrogen 2 atm. This goes 2 times. So we get E naught is 0 anyways for this oxidation, 0.03 times. This will be log 2 plus log 10 power minus 4, which will be minus 4 plus 0.3, minus 3.70. Okay. And now you can do the calculation. 3 times 3.7 is going to be 10111, 111 it will be. So minus minus plus 0.111 volt around, okay, which is going to be first option 109, which is mentioned here. Okay, cutoff. Miss is asking for the cutoff. Cutoff would be predicted by our favorite Gandhi sir just after this paper discussion. So I don't know how physics paper is till now. After all the discussions are done, then Gandhi sir will come and tell you that what would be the expected cutoff for each section, which is going to be for unreserved and reserved category as well. Okay, right. So we'll not jump on to the conclusion that cutoff will be high or low yet. We'll wait for the final discussion. Okay. Question number 56. It's a very simple question. I hope it doesn't require any of your discussion. Zero order means same as the rate units, which is mole per liter per second. First order, time minus 1. Second order, molarity minus 1, time minus 1. And third order, molarity minus 2, time minus 1, which is going to be molarity to the power 1 minus order, second ki power minus 1. This will be the units. N is the order. Okay. Moving ahead, titanium plus 2, and see how much easier they can make a question. They gave you the configuration as well. In general, they shouldn't give the configuration. It should be the work of student to not clearly audible. Am I not clearly audible? I'll just shift the mic little up. Can you confirm, is it audible now? Just a second. Is it audible now? Okay. So 2 unpaired electron 3d2, which is going to be root of 2 into 2 plus 2 Bohr magneton. Okay. So I hope all of you will be able to get his root 8, which is 2.84 Bohr magneton. Okay. Moving to the next one. Question number 58. First reaction which we are doing here is Friedel-Crafts alkylation to get a toluene. And then we are doing nitration. Both of the reactions are same reactions which are going to electrophilic substitution. Minor product will be orthonitro and major product will be paranitro, as CH3 is a ring activating ortho-para directing group. Okay. Now, how do we separate them? How do we separate them? Continuous extraction, differential extraction cannot be used here, because for this one has to be less polar than other. One should be more soluble in water, one should be more soluble in benzene. But both the compounds are organic compounds. So obviously differential extraction will not work here. Sublimation also will not work here because these two are liquids in nature. And because they are very similar in the shape and structure, will require a fractional distillation because their boiling point should be very close to each other. Okay. You have seen the example of orthonitrophenol and paranitrophenol where you separate them by steam distillation. So if nitrophenols are done by distillation, these are going to have even lesser force of attraction than nitrophenols. They have hydrogen bonding. If they are liquids, they will be even more volatile liquids. Both of them will be liquids, volatile liquids, so you can do the distillation. Fractional distillation would be the answer here. Next question is, a bulb is rated 150 watt, 8% of the energy is converted into light. So first we should think the bulb is actually not 150 watts. The working power is only 8% of 150. So just remove this power. This is useless power for us. The power which gets converted into energy is only 12 watts, which is 8% of 150. 8% of 150. Power equal to energy by time. Time is given 1 second. So energy in joules come out to be 12 joules. Okay. And we know the Planck's quantum theory, energy equal to n time h nu, which is energy of each photon. So number of photons into energy of each photon, which is given to you directly, 4.4 10 power minus 19. 4.4 10 power minus 19. So from here, number of photons they are asking, which comes out to be 12 by 4.4 into 10 power plus 19. This obviously goes almost at 3, less than 3, 2 point something, which is going to be 2.7 into 10 power 19. There is no other option nearby. So directly we can choose the third option. Right? I hope it is clear to everyone up to here. Moving ahead to question number 60. In the test tube containing a salt, few drops of H2SO4 is added, which gave a colorless vapor which have a smell of vinegar. Although in your practical chemistry, acetate ion is not mentioned in the syllabus, but all of you know what is vinegar, and you can easily choose from here acetate ion. Acetate converts to acetic acid, which will give you the vinegar smell. Will do. Okay. Reduction of nitriles means cyanides to primary amine. It's a direct question from the NCERT reagents. LiLH4 we know can be used. SnHCl cannot be used. SnHCl is actually used for nitro to amines, not from cyanides to amines. Okay, so SnHCl will not be used here. Hydrogen nickel will work here as we know. Sodium amalgam ethanol is written directly in the NCERT. So yes, we have to use A, C or D, which is going to be option 2, ACD. ACD can be used. Any of them can be used for this conversion. Next is question number 62. Incorrect statement they are asking. Carbon has the ability to form p pi p pi. Yes, it is correct only. It's a correct statement. In alkenes and alkynes, you see carbon forms p pi multiple bond. It is a monomer when it's BCl3, which is correct due to back bonding. It doesn't undergo any dimerization. AlCl3 undergoes dimerization, we know. So it's also a correct statement. Oxygen exhibit only -2. This is incorrect. Okay, obviously oxygen can show peroxide, oxygen can show +1 also in O2F2, +2 also in OF2. Why cerium shows +4 oxidation state but others show +3? Because cerium +4 is a noble gas configuration. Cerium +4 is going to be a noble gas configuration. Its nearest inert gas is radon. It doesn't explain anything. Everyone will have some nearest inert gas, but doesn't explain why it is stable. It has a noble gas configuration. After losing one more electron, it requires 4F14. That is not correct. Okay, 4F14 it will not acquire. 4F14 will be acquired by the last elements, which is going to be lutetium. Its atomic number is 61. Doesn't explain anything. Okay, although it's not 61 as well, it's 58 cerium. After losing one more electron, it becomes 4F0, which is a noble gas configuration, which is correct. 64. During Lazine's test, what happens? Covalent bonds are converted into ionic form. So all the nitrogen which is present in, for example, aniline, all these nitrogen from the covalently bonded nitrogen will be converted to sodium cyanide. So we have prepared an inorganic compound from the organic compound. We have converted our covalent bonds into ionic compounds. Okay. Was chemistry easy? I would say it was easy, barring 2 to 3 questions. The rest of the questions were easy only. Mostly NCERT based. So if you are well versed with the NCERT, you wouldn't do many mistakes. How many H atoms are present in urea? Urea, NH2, CONH2. Okay, 5.4 gram of urea is taken. So we can say moles of urea are going to be 5.4 by 60 gram, which is the molar mass of urea. These are the moles of urea. Molecules of urea are going to be 5.4 by 60 moles into Avogadro number. These many molecules of urea are present. And each urea has 4 hydrogens. So number of hydrogen atoms will be 4 times the number of urea molecules, which is going to be 4 times this number. 5.4 by 60 into 6 into 10 power 23. Okay, this go 10 times, this is 10 power 22. So you get it as 21.6 into 10 power 22. Okay, which is going to be 2.16 10 power 23. Okay, 10 power 23. Right? All right. Moving to the next one. Question number 66. Metamers. Very famous question and very easy question. Some of you might have marked this one. It's not correct. These are not both ketones. One of them is an aldehyde, so they are functional isomers, not metamers. If both were ketones and the position was different, they could have been called metamers, like pentane 2 on pentane 3 on, but these are not metamers. Obviously, ethers can easily show metamerism. Most common example of metamerism is this only. Question coming from group 15. PC2H5 whole thrice, AsC6H5 whole thrice, triphenyl arsenine, triethyl phosphine can form d-pi multiple bonding with the transition metal. Yes, very correct statement. We call it as synergic bonding. So if a nickel plus 2 is bonding with PPH3, for example, okay, for example this, then as you have seen synergic bonding in nickel carbonyls, here also phosphorus has a vacant d orbital, 3d. Nickel has a fully filled 3d orbital. By back bonding, metal can make good synergy with the phosphorus. So that's why triphenyl phosphines are strong ligands. Trimethyl phosphine, triphenyl phosphine. Yes, it is correct statement. Directly written in the anomalous properties of nitrogen, that nitrogen can't do this, but others can. Nitrogen can form d-pi p-pi bond, can never do that. Nitrogen doesn't have a d orbital, so second statement is incorrect. Next, phenolphthalein is used as an indicator for sodium hydroxide against oxalic acid. The color change that is observed at an alkaline pH means when you reach to higher pH, what will happen? Like this, the graph will come, if you remember. Okay. Near to the alkaline pH, here phenolphthalein works from 8.5 to 10.5. As soon as you reach to alkaline pH, it will become pink color. So it will go from colorless to pink. It will go from colorless to pink, which is going to be third option. Colorless to pink. Okay. Because they mentioned alkaline, means we have to see going to the higher pH what the color becomes. This one is a direct question. C2H2 ethine has 2 pi bonds, as you know. There is no other option of 2 pi bonds. So B has to match with 1. There is only one option there. So we will not check the other ones. At a certain temperature during a process, 500 joule heat is absorbed by the system. Work is done by the system. Delta U equal to Q plus W. Q is absorbed, so plus 500. Work is done by the system means its expansion. Work will be negative. Minus 300. Minus 200. So which is going to be plus 300, the delta U. Okay, again a very simple question. Again NCERT equation, methane reacts with steam to give syngas, CO plus hydrogen. Gives you syngas, CO plus hydrogen, CH4 plus steam. Gives you CO plus H2. CO plus 3 moles H2 you can make here. Reaction with steam in the presence of nickel catalyst, 1273 Kelvin. Again, directly NCERT reaction that they have given. Almost, I would say 95% of the paper is directly from the NCERT. This particular question is the only one which is little dicey, which has 2 correct options possible here. Okay. It's also from the NCERT. Direct question is coming from the NCERT in text question. In the aldehyde ketone chapter. Compound C8H8O gives red-orange precipitate, means Brady's reagent test. This will also give, this will also give, this will also give, this will also give. Every aldehyde and every ketone gives the Brady's reagent test. It does not reduce Fehling reagent. It will also not reduce because ketones can't, ketones cannot, aromatic aldehyde cannot react with Fehling. So all options are right up to here. On drastic oxidation, Okay, drastic oxidation. We know that ketones can be oxidized drastically at drastic conditions with KMNO4 or chromic acid to convert to carboxylic acid. It's possible here, it's possible here, it's possible at methylated benzene as well. Okay, toluene to benzoic acid, also NCERT writes, it is oxidized with KMNO4 KOH heating or chromic acid at vigorous oxidation, they have mentioned, if you look at the NCERT. So, this is also right. Under vigorous condition, CH3 can be oxidized to COH. So, till now all the options are coming correct. Gives P, which is an aromatic product. Let's see. This is to anyways not possible because it's never the alpha carbon which gets oxidized. It has to break from here and become carboxylic acid. So this is not a possible option. This is possible. A ketone can be oxidized to carboxylic acid. This is absolutely a correct answer. Okay, I would say this is the perfect key because NCERT has given the same reaction. It's a perfect key. 2 is correct. Okay. Coming to third one is also absolutely correct. There is no problem in the third option as well. Both 2 and 3 are fine. But in the first time when the key will be produced, most probably you will get second answer, okay, instead of third. Third, there is no problem. NCERT writes nicely that alkylated benzenes under vigorous oxidation with chromic acid give benzoic acid. So this is also correct, where aldehyde will be oxidized easily and this will require the drastic condition. And both of them are aromatic compound. This is also aromatic. This so both 2 and 3 are correct. This is not correct because CH3 will not remain CH3 after oxidation. So 2 and 3 both are correct here. This is the only question in chemistry which might create a issue. But other than that, other questions have the direct answers. These have 2 answers. Better one would be still second option because it's directly coming from the NCERT in text. Both are correct. A solution of copper sulfate is electrolyzed for 10 minutes using current of 1.5 ampere. Mass of copper deposited. You can use Faraday's first law directly. Weight of copper deposited equal to E by FIT. Equivalent weight of copper, which is going to be 63 gram by 2 because copper is coming from +2 oxidation state. So equivalent mass will be molar mass by n factor 2. Faraday 96500. Current is 1.5 ampere. Time is 600 seconds. Okay, time always will be taken in seconds. So this is our weight of copper obtained from here, which you can solve easily and you will get it as 0.3 gram around. 0.29 grams. Directly you will be getting it. Since the options look very far away from each other, you can do a lot of approximation. There is no problem. If you see the options, they are very far away from each other. Don't have to get the very correct answer. You can just do approximation, like on the table of 6 or 3, for example, I can cut it at 21 times 3, 2, 1. Okay, 3, 2, 1, 6 times 3, 2, 1, 6, 6 times. Almost take it 3200 if you wish. Okay, and then this is 300. Again, in the table of 3, I can cut it at 100. And this is going at 10722 around 10722. Okay, and then this is 100. This is 107 almost at the bottom. Okay, again on the table of 3, 7 times, and this is 39 times. So we get it as 15 by 390. This is almost 400 if you consider it, which will be around 0.3. Okay, or you can just do the calculation part. It won't take much time. This is about 73. Next is phthalene dye test. Phthalene dye test, which is nothing but phenolphthalein preparation reaction. We know that when 2 moles of phenol react with phthalic anhydride, you get a dye which is called as phenolphthalein, and on putting NaOH, it will turn pink in color. That is what the test is, phthalene dye test. Which compound will react with phthalic anhydride to give you a dye which gives pink color in NaOH. That is the test. So phenols will be reacting there, as we know. Phenols will be reacting there. Phthalene dye test, where you get phenolphthalein as the final product. It's a 2-time electrophilic substitution you are doing at the para position of phenol. Like this. This way is colorless, but when you put NaOH, this OH loses the proton, changes into quininoid form, which will be pink in color. You can check that it is given in the practical chemistry part of the NCRT. Okay, so it is going to be phenolic groups which can give you the phthalene dye test. Other tests for phenolic group also you have studied, neutral FeCl3 test and bromine water test and so on. This is your again biology type question. DNA, RNA, what does it contain? DNA is double stranded and it has ATGC, so thymine is one of the base available. Okay. Coming to question number 76. Choose the best option here. I would suggest first to require little bit of, checking the calculation, but A is there in all the options, so I don't have to check A. A would be anyways correct. B and C. B is again, you have to check for the value. I would suggest to leave it and go for the C. Aquatic species are more comfortable in cold water, which is absolutely correct. Solubility of oxygen will be more in the cold condition. So gases are more soluble at low temperature and aquatic species will be more comfortable. C is correct. A is correct, C is correct. So now you are at 2 options. Now you can check for the B because only one option I need to check now whether B is correct or not. Molarity of a solution containing 5 gram NaOH. So molarity will be 5 gram NaOH by molar mass 40 gram into 1000 by 450 ml of 450 ml of the solution volume. When you solve it, you are getting exactly 0.278 only. So it's a correct statement. A, B, C all are correct here. Okay, A, B, C all are correct here. Question number 77. Negative deviation comes. Why is there a negative deviation? Because solute solvent particles have better interaction because of hydrogen bonding. A very nice NCRT line to line question is being asked. This diagram, if you would have seen in the NCRT, due to hydrogen bonding between chloroform and acetone, it shows negative deviation. Other all options are for positive deviation there. A certain buffer solution contain equal concentration of X minus and HX. Conjugate base and acid both are equal. Kb of the X minus conjugate base is given 10 power minus 10. We know that pKb is 10. So pKa of HX would be 14 minus pKb, which will be 4. And this is exactly half equivalent point where both salt and acid are equal. pH of the acidic buffer will be pKa plus log equal 1 by 1, which is log 1 is 0. So pH will be pKa only, which is 4. Half neutralization point. Right? Question number 79, again a very easy question. Looking at the second option, largest and the smallest species. We know that among Mg plus 2 and Al plus 3, Al plus 3 will be smaller in size because isoelectronic species. Isoelectronic species, more positive charge, more Z effective, smaller the size. Hi Hassan. Okay. And among aluminium and magnesium, left to right size decreases. So aluminium should be smaller, magnesium should be largest. So largest should be Mg, smallest should be Al plus 3. They are saying the largest is Mg, which is correct. Smallest is Mg plus 2, that is not correct. Achha, they are saying largest is Al, which is not correct. And the smallest is Mg plus 2, which is also not correct. So both are not correct here and they are asking incorrect statement only. Okay, so this is the one. Question 80 is on the value of electronegativity. Sodium, as we know, 0.9, magnesium 1.2, beryllium 1.5, silicon 1.8, phosphorus 2.1. Electronegativity values have to be remembered. Lesser the value, more the metallic character. Or you can look at their respective positions as well. Sodium is here, beryllium is here, magnesium is here, and then silicon is present somewhere here in the line of magnesium only, group 14. And then phosphorus is present here in the 15th group. We know that top to bottom metallic nature increases. So on left to right, metallic nature decreases. So on the most left side, this is going to be most metallic. This is going to be least metallic. So middle, so we'll have P at the last, Na on the top. P at the last, Na on the top. Two options are coming from here. You can easily compare beryllium, magnesium. Top to bottom, metallic character is more. So this is better than this one. So magnesium should be better than beryllium. So this is wrong. This is right. Okay. Again, a NCERT question where they are asking for the lowest locant rule. Okay. Lowest locant rule is not applicable here because both sides you are getting locant 3 and 5, 3 and 5. Then prefix rule we have to see whichever substituent. Thank you, Ayushi Agarwal. So whichever substituent has alphabetically prior name, which is ethyl here, E, this is methyl here, M, E will be given more preference, will be giving 3 here, 3 ethyl, 5 methyl heptane. Same to same NCERT in text question. Again NCERT direct question. Platinum with all complexes with coordination number 4 are going to be square plane. Anything with 6 coordination has to be octahedral. So 3 and 1, 3 and 1, only one option possible. We'll not check for the others then. 83. Concentration changes with time as a linear. AT equal to A0 minus KT, which is zero order reaction. Zero order reaction. Okay. 84. Which is a ambidentate ligand? We know SCN negative is ambidentate ligand. It can attach from sulfur side also, nitrogen side also. Next question. Delta U naught is given minus 10 kilojoule. Delta S is given minus 44 joules at 300 Kelvin almost. What is delta G naught? Okay. So we'll use the Gibbs-Helmholtz equation, which is delta G naught equal to delta H naught minus T delta S naught. Delta H is not given, instead delta U is given. So I'll put the value as delta U minus P delta V delta NGRT. Since equation is given here, delta NGRT will mention delta H equal to delta U plus delta NGRT, sorry, minus T delta S. Minus T delta S. So delta G naught we have to find, just put in the values now. Delta U is minus 10, so I'll put it as minus 10,000 joules. Plus delta NGRT. Delta NG if you see is minus 1 here. So plus minus 1 into R. In joules R is 25 by 3, which is 8.3. Into temperature, although 298 given, I can approximate it to 300 because anyways options are very far. There is no option very close, only if plus minus if I get, I'll be able to solve it. Minus T delta S, again temperature 300, delta S is minus 44. Everything is taken in joules, so we'll get the answer in joules only. Minus 10,000 minus 2,500 means minus 12,500. Minus minus plus 44 into 3 is 132, 132,000. So minus 12,000 plus 13,000, answer is coming positive 700 joules. Positive 700 means plus 0.635 kilojoule, and it is going to be non-spontaneous. Non-spontaneous, as delta G is coming positive, we'll call it non-spontaneous. Question 86, again from the NCERT. Line to line questions have come from NCERT this year. This oxygen will have 0 formal charge. I hope all of you know how to find the formal charge, plus 1 and minus 1. 0, plus 1 and minus 1 are the formal charges on the given number of oxygen, 2, 1 and 3. Right? 87. In which of the following Kp is not equal to Kc? We know Kp equal to Kc into RT to the power delta Ng. If delta Ng is 0, then only Kp and Kc are equal. If delta Ng is 0, then only Kp and Kc are equal. So here delta Ng is 0, Kp and Kc are equal. Delta Ng is 0, Kp and Kc are equal. Delta Ng is minus 2, 2 minus 4 minus 2. So here they will be different. Delta Ng is negative here, so Kp will be less than Kc here. Question 88. This coming from your Arrhenius equation, which says ln k equal to ln A minus Ea by RT. So this term is ln A obviously, and this term is Ea by R. This term is Ea by R. This term is Ea by R because T is separate only. So Ea by R value is 1.25 into 10 power 4. They are asking in kilocalorie. That means this value is given in calorie. Okay. If I take R as 2 calorie, then Ea comes out to be 2 times this, which is 2.5 into 10 power 4 calorie. Or I can say 25 kilocalorie, which is directly option 4. Okay. So this is question 88. 2 more we have. Let us quickly solve them. Yes. C2H5Cl with some X gives Z. Okay. Then this compound, which is propanamide, on Hofmann bromide degradation, CO will go out as we know. It's a reaction with a step down. We get a 1 carbon less amine, which is going to be this, which is mentioned here, correct? Ethanamine. Ethanamine, this is carbyl amine reaction. Carbyl amine reaction, bad smelling compound, we get isocyanide. We get isocyanide here, isocyanide. Okay, so Z is this. Here also Z is this, that means who should react with the alkyl halide to give isocyanide? It should be AgCN. Because KCN produces cyanides, AgCN produces isocyanide. So AgCN and the isocyanide would be your answer, 4th option. Moving to the last one, question number 90. Again a simple question coming directly from the NCERT isomerism example. None of the question is outside of the NCERT today. Cisplatin, transplatin, GI. Cisplatin used for anti-cancer treatment. COEN thrice means MAA whole thrice. 3 rings are there. As we know, when 3 rings are present on the octahedral, it is always going to be optically active. There is no plane of symmetry possible here. 1 ring will be here, 1 ring will be here, 1 ring will be here. It will be dextro-levo 2 option. So optical isomerism. It is a MA5B type. It can't show GI or OI, but NO2 can show ONO. So linkage it can show ionization as well. There is no option of ionization, so linkage. And obviously water is there, so hydrate isomerism can be there, which is solvate isomerism, same meaning. Okay, so I hope you all have attempted your exam well. Okay, all the very best for your result of NEET. And now we'll be going on with the discussion of physics with our esteemed physics faculties. So I'll be taking your leave now. All the best. Bye bye. See you all.
Hi students, so I hope that all of you have given your exam very well. So now we are going to discuss the physics paper, and before starting the paper, just like the paper which came before that, speculations were rife that 2025 paper, it was say moderate or tough, so many were expecting and giving predictions that this year also the physics is going to be very tough. But it came as a surprise because the questions, I hope you have seen them and you understood that they were very easy, barring a few or two, means 2 or 3 questions where might be there would be some ambiguity in the language or something, but overall the paper was doable. 1 or 2 numerical calculations, I would accept that yes, they were a bit lengthy, that too, but still you had lot of scope of attempting the questions. They were very easy questions, right? So we are going to see the questions. So I'll be discussing the first 9 questions over here. Let's see them one by one. So we have the first question where 100 turn closely wound circular coil of radius 5 centimeter has a magnetic field of 3.14 into 10 to the power minus 3. So they have given the data, and magnetic field at the center, which we know is mu naught Ni upon 2R. So they have given the value of magnetic field as 3.14, which we can take as pi into 10 to the power minus 3 in SI units. Rest, they are asking what? The magnitude of the magnetic moment of the coil and also the current flowing. So here the radius of the coil has been given as 5 centimeter, which you can write as 5 into 10 to the power minus 2 in SI units. Now apart from that, you know very well mu naught can be written as 4 pi into 10 to the power minus of 7 SI units. Number of turns given 100, we have to just find the value of I. So you know from here that pi cancels, and here 5 2s are 10. So an easy calculation gives you 5 by 2, which is 2.5. So 2.5 amperes is going to be the current flowing in the coil, right? Now they have also asked the magnitude of the magnetic moment. So for a loop, we know magnetic moment is current into area of the loop. There are number of turns given. So NIA, area of the coil or single loop, will be pi r square. Again, just put the value of pi as, sorry, 5 radius as 5 into 10 to the power minus 2, and number of turns 100, and current is given as 2.5. So just putting the values over here, 100, 2.55, and so you'll get this value closely. It will come as 2, because pi you have to take as 3.14, so 20. So this is the magnetic moment. Now moving on to the next question. Next question, we have match the column. We have been given list 1 and list 2, and here we have to match, I think. Okay, E is equals to h nu. E equals to h nu, we know, is the energy of a photon. Energy of a photon, we have A matching with 4. And second one is diffraction and interference. These show the or exhibit the wave nature of light. So diffraction and interference, we will see, is the third one. And lambda is equals to h by p, which shows the de Broglie wavelength. And de Broglie wavelength we have first. And next we have Compton effect. Compton effect simply shows the particle nature of light, which is second. So we can go for a perfect match from this. Now next question. The current I in the circuit shown below is, okay, all the diodes are ideal and identical. So we have this 10 volt battery and we have 4 parallel branches. They are consisting of diodes and they are in parallel. Now here you can see, as per the polarity, this diode becomes reverse biased because it will try to send current like this. So this is reverse biased. This one is forward biased. This one again is reverse biased, and this one becomes forward biased. So from here, the reverse biased diodes will be simply removed. Those branches will be removed. We have 4 ohm and 2 ohm left in parallel. So we have R equivalent of the circuit is nothing but 4 ohm parallel with 2 ohm, which will become 4 2s are 8 by 6, that is 4 by 3 ohm. So they are asking the value of I. So current is EMF divided by R equivalent. They have given the value of EMF as 10 volts. So 10 by 4 by 3, which is 30 by 4. They have given it in fraction itself, 15 by 2. So 15 by 2 ampere will be the correct answer for this question, which I think second option is marked already. I hope it is clear. 2 reverse bias diodes are removed. Next we come to the fourth question, wherein the speed of light in vacuum is taken as unity. So they are considering 3 into 10 to the power 8 meter per second as equivalent to 1 unit of speed. So they are asking, if it is 1 unit, then how much is the distance between sun and earth? Now distance we can write as speed of light into time. So since you are taking already V as 1 unit, so you have to focus only on time, and in time you have to convert in seconds. Why seconds? Mind it, over here you can see second coming. So here T is given as 6 minutes 40 seconds. 6 minutes means 6 into 60 and plus 40, which is 360 plus 40, 400 seconds. So we can say the value of D will be 400 units. So 400, which is the third option. I hope this too is clear to all of you. Okay, let's move on. Next, we have fifth question. The following plots show the variation of velocity with time of a ball thrown vertically upward and falling back. Which of the following plots is or are correct? Now, if you see in this particular question, we'll just follow the convention generally used, that when we talk of motion, we take the initial direction of motion as positive. So where are we throwing it? Upward. So we are simply going to take the upward direction as positive, right? Now, throughout motion under gravity, you know what is the acceleration? Acceleration will be g downwards. Now, since it is g downwards, can I take acceleration as minus g? Minus g means it is a negative constant. Now, since it is negative constant, slope of VT graph is negative constant. Means what? VT graph is a decreasing straight line. So, decreasing straight line, which one is that? We can see here C option, decreasing straight line. Acceleration is slope of VT graph. It is a negative constant. So, VT graph is a decreasing straight line. So, which option shall we select for this? C only, which is given in which one? First option. Okay. Coming to next question, in a vernier calipers, 20 vernier scale divisions coincide with 16 main scale divisions, and each division of main scale is given as 1 mm. So they are asking the least count of the vernier calipers. So just going by the expression, least count of vernier calipers is nothing but it is the difference of least count of main scale and least count of vernier scale, which is simply 1 MSD minus 1 VSD. This is what we do, and 1 MSD is given to us as how much? 1 mm. And what is 1 VSD? They have given 20 VSD equals 16 MSD. So 1 VSD will become 16 by 20 MSD. 16 by 20 MSD, and 1 MSD is how much? 1 mm. Got it? 16 by 20 mm. So 1 minus 16 by 20, that is 4 by 20. 4 by 20 means 1 by 5 mm. So we can say it is 0.2 mm, or if you convert, they have given the units in centimeters. So 0.2 mm can be written as 0.02 centimeter. That means we should go with option 3. Clear? Next question, 7th. An AC circuit contains a resistance of 1 kilo ohm, capacitor of 0.1 microfarad, and an inductor of 1 millihenry connected in series. The resonance frequency they are asking approximately. Now, when it is at resonance, we know that resonant frequency is independent of resistance. It depends on the value of L and C given. So, what is omega? The resonant frequency omega naught we know is 1 by root LC, and omega naught can also be written as 2 pi into frequency. They are asking F naught. Okay. So, what we simply do? The value of inductance they have given 1 millihenry, that is 10 to the power minus 3 SI units. C, capacitance they have given 0.1 microfarad. 0.1 microfarad means 10 to the power minus 6, and you have one more 0.1 which makes it 10 to the power minus 7. So from here 10 to the power minus 10 will come. Under root when you take it becomes 10 to the power minus 5. It goes up, becomes 10 to the power 5. So 2 pi f naught is equals to 10 to the power 5. So they are asking us the value of f naught. Approximately, if I had to do this calculation, what I will do, 10 we are seeing from here. So 10 by 2 becomes 5, and we have 10 more if we take, then we have 10 to the power 3, and what else you are having below is pi. This is what you are taking. Now 10 to the power 3 will make it kilohertz, right? 10 to the power 3 will make it kilohertz. Let's see this calculation. We know that pi square is approximately 10. So 10 value you can approximately take as pi squared. So this will approximately become 5, 1 pi cancels, 5 pi. So 5 pi kilohertz. So if you take the value of pi as 3.14, so 5 3s are 15 kilohertz, closest answer to 15 kilohertz we'll see is 15.9 kilohertz. So we'll say that approximately it will be 15.9 kilohertz. Clear? So just going by this formula, and we can avoid lengthy calculations just by taking 10 as pi square, we'll get the answer. Now here, next question we have, 8th one. We have a long straight solid wire of circular cross section radius A carrying steady current I. Current I is uniformly distributed through the whole cross section. They are asking the variation of magnetic field. It is a direct formula based question where we have inside, when we take internal point, you have studied mu naught I by 2 pi r squared into small r. Here we have to take capital R, they have taken as the radius as small a, and small r is the radial distance. So it is directly proportional to r when we have internal point. And when we have external point, we go with same formula like a thin wire. So we have mu naught I by 2 pi r, which is inversely proportional to r. So inside we have linear variation, straight line passing through origin, and outside we have a rectangular hyperbola. So you can see which option matches the first one. So inside you have directly proportional to r, and outside we have inversely proportional to r, which is a rectangular hyperbola. So first option will be the correct one. So coming to our 9th question, we have a uniform metallic wire having resistance of 4 ohm. So it is bent to form a square loop. This is a square loop and they have given 4 ohm. That means each arm we can say will be of 1 ohm. Now if you see this point A and C, we have connected it to a battery, and there is a resistor attached between the terminals B and D, which clearly gives an indication of a balanced Wheatstone bridge between the points A and C. Okay, so what happens, 2 ohm can be simply removed. We can remove the 2 ohm. So they are asking what is the value of this current I. So simply if you remove this, you can see we have just 1 ohm will come in series, will become 2 ohm. 1 ohm will come in series, 2 ohm between the points A and C. Now 2 ohm are in parallel, so 2 by 2, that is 1 ohm. So effective resistance of the circuit between the points A and C becomes 1 ohm, and you have an EMF of 2 volt, which clearly tells us the value of current is going to be EMF by the equivalent resistance, which is how much? 2 ampere. So we are done with this. We have seen that first option we are getting for this particular question. So I hope the concepts what we have discussed over here, you understood this. I hope it is clear to all of you. So after me, other teachers will continue with the discussion of the questions from 10th onwards. Okay, so thank you.
Hello students, so I hope that all the questions were clear to you. So 9 questions have been done. Now we will be recording the remaining questions. So I'll be recording the 10 to 19 or 10 to 18 questions I'll be recording. So let's start the question. So the question number 10 is that they are saying that there is a unknown nucleus and it is a nuclear density of this much, and they have given the mass of the nucleus as this much. They are asking you what is the mass number A approximately. See here, there is one logic of doing this particular question completely in detail. So let me tell you the approach also first, then we'll look at the approximation which we can do. We can easily solve this question without doing all the calculations also. So let's first discuss the approach. The approach is that the density of the material is given to you, and density can be written as mass divided by volume. And we very well know that volume can be written as 4 by 3 pi r cube. So it will be mass divided by 4 by 3 pi r cube. R can be written as what? R can be written as r naught into a to the power 1 by 3. We all know this. So what we'll get is, we'll get r naught cube into multiplied by a. Now everything is given to us. Density also is given to us, mass also is given to us, r naught value is also given to us, the value of 4 pi is also given to us, and from here we can get the value of mass number A. Okay? From here we can get the value of mass number A. That A will come out to be something in kg, it will come out to be. We can do the calculations, it will come out to be in kg. And from that particular mass number we can convert it into AMU. We very well know that 1 AMU is given by 1.67 into 10 to the power minus 27 kg. So 1 kg will be equals to what? 1 kg will be equals to 1 divided by 1.67 into 10 to the power minus 27 AMU. So once we get the value of mass number in kg, we will divide it by 1.67 into 10 to the power minus 27, we'll get it in AMU. But let me tell you, there is no need to do all these calculations. If your presence of mind is good, then you can look at the data. The data is saying that it is 19.926 into 10 to the power minus 27 kg. And the options are given like this. Now if you see 10 AMU, I already told you that 1 AMU is this much, 1.67 10 to the power minus 27. 10 AMU will be how much? 10 AMU will be multiplied by 10. It will be 16.7 into 10 to the power minus 27 kg. Now if you look at the options, 12 AMU is given, 19 is given, 20 is given, and 16 is given. These options are very far away from this particular value, because 10 AMU is 16.7 and 19.926 is approximately close to 16.7. So if you go by that particular approach, by that approximation, then directly you can take that option number 1 will be the correct answer. Even if you solve for 16 also, you will do a simple calculation of 16 multiplied by this, and then you will get the answer which will be more than this 9.926. So from this particular logic, you can directly take option number 1 and you can save a lot of time in this question. Otherwise, you have to do all these calculations and you have to get the answer. So this was a very good approach. By this presence of mind, you can do this particular question. So let's move on to the next question. So the correct answer for this is option number 1. Next question is, they are saying that there is a rectangular wire loop of sides 8 cm and 3 cm with a small cut. Since the rectangular loop is having a small cut, so obviously the current cannot flow, but an EMF can be induced in this particular loop because it is moving out of a region of uniform magnetic field of magnitude 0.3 Tesla. So the magnetic field is given to you as 0.3 Tesla, and it is directed normal to the plane of the loop. So magnetic field, we can assume that if this is the rectangular loop, suppose there is a small cut here, and this is the magnetic field. So inside magnetic field is there perpendicular to the plane of the loop, and this particular loop is coming out of the magnetic field. This length is given to you as 8 cm and this length is given to you as 3 cm. And they have said that the velocity of the loop is 2 cm per second in a direction normal to the shorter length of the loop. So the shorter length is this, normal or perpendicular to this particular loop or wire, the velocity is given as 2 cm per second. So basically, if I will calculate the EMF, see, current I cannot calculate, there cannot be current because it is an open loop, but EMF can be developed and EMF will be given by BVL. Now which length we have to take? We have to take the length which is perpendicular to this velocity. So 3 cm is perpendicular to the velocity, we cannot take 8 cm. So from there we will get the answer. So 0.3 multiplied by 2 cm it is, to convert it into meter, 2 by 100, multiplied by length is 3 cm, converted it will be 3 by 100, and what you will get from here, you will get 3 into 2 6, 6 into 3 is 18, so 1.8 into 10 to the power minus 4 volt. So if we look at the options, 1.8 into 10 to the power minus 4 volt, option number 1 will be the correct answer. I hope it is clear to you. Let's move to the next question. It is saying that a galvanometer of resistance 100 ohm gives full scale deflection for a current of 1 milliampere. So The resistance of the galvanometer is given to you. Resistance of the galvanometer is 100 ohm. And the full scale deflection current is also given to you, and that full scale deflection current is Ig, and that is basically equal to 1 milliampere. We can also write it as 10 to the power minus 3 ampere. Now they are saying that it is converted into an ammeter of range 0 to 10 ampere. So the total range of that ammeter is 10 ampere. So what I can do is I can draw the diagram that it will consist of the galvanometer, and then there will be a shunt which will be in the parallel, and the current which is entering from here, the maximum current which can enter from here will be 10 ampere. Out of that, the Ig which is going here will be equals to 10 to the power minus 3 ampere, which is 1 milliampere. So the remaining part will be going from here, that will be 10 minus Ig. Now we very well know that across this particular two resistors, they are in parallel, so potential difference will be same. So I can write that Ig multiplied by Rg can be written as S multiplied by 10 minus Ig. They are asking you the value of shunt required, so from here we can get the value of shunt. Very simple, Ig will be 10 to the power minus 3 multiplied by the resistance of the galvanometer is 10 to the power 2 is equal to shunt into. Now if you see properly, since they are asking approximate value, Ig is very much less. Ig is of the order of 10 to the power minus 3 ampere, which is negligible compared to this value 10. So I can approximately equate it to 10. So what I am getting, shunt will be equals to 10 to the power minus 3 multiplied by 10. It will be 10 to the power minus 2 ohm, which will give me 0.01 ohm as the answer. So the correct answer for this question will be option number 1. I hope it is clear to you. So let's move to the next question. Question number 13. In the question number 13, what they are saying is that in Young's double slit experiment, using monochromatic light of wavelength lambda, the intensity of light at a point on the screen where the part difference is lambda is k units. See, since they have not given the individual sources intensity, so we will assume that it is equal. So let us take the individual intensity, individual source intensity, let us take it as I0. So what will be the maximum intensity? The maximum intensity will be 4I0, we very well know. And we know the general result also, that the general intensity can be given as 4I0 cos square delta phi divided by 2, where delta phi is basically the phase difference. Now this maximum intensity is given to you as k units because it is the intensity when part difference is lambda, and for part difference lambda we know very well that it is the constructive interference, maximum will be there, so the intensity will be maximum. So 4I0 is given to you as k units, and they are asking find the intensity of light at a point where the part difference is lambda by 3. So since the part difference is lambda by 3, let me calculate the phase difference. Phase difference will be 2 pi by lambda multiplied by delta x lambda by 3, so it will come out to be 2 pi by 3. So from here, if I will calculate the intensity, I will get it as 4I0 cos square 2 pi by 3, or I can write like this, cos 2 pi by 3 divided by 2 whole square. This will come out to be cos pi by 3. Cos pi by 3 is basically cos 60 degree 1 by 2. 1 by 2 whole square will be 4. So the intensity is coming out to be I0. But I have to give the answer in terms of k. I know that 4I0 is equal to k. So I0 will be equal to how much? It will be k by 4. So the correct answer for this question will be option number 1. I hope it is clear to you. Let's move to the next question. The magnitude and direction of the acceleration produced in a body of mass 5 kg. So there is a body of mass 5 kg and two mutually perpendicular forces are acting. One is 8 Newton and one is 6 Newton. They are asking you that what is the acceleration of the body. So let's calculate the net force acting on the body. The net force acting on the body will be equal to how much? We very well know, this is very simple. Net force will be under root of 8 square plus 6 square. This will come out to be 10 Newton. The magnitude of acceleration will be net force divided by mass. 10 divided by 5, that will be 2 meter per second square. So 2 meter per second square is the magnitude, but they are asking you the direction also. So what we can do is, we can take this angle as theta. Tan theta, if you will calculate, tan theta will be equals to 6 divided by 8. 6 divided by 8 is 3 by 4. So theta will be equals to what? Tan inverse 3 by 4. And this angle which the net force is making or the acceleration is making, it is making with the 8 Newton force. So the angle with which this particular force is making the direction with 8 Newton will be tan inverse of 3 by 4. So the correct answer will be option number 3, that the acceleration magnitude is 2 meter per second square and it is at an angle of tan inverse 3 by 4 with 8 Newton force. So option number 3 will be the correct answer. Let's move to the next question. 5 capacitors are there, 4 are connected in series. If you look properly, C1, C2, C3, C4 are in series. So what will be the equivalent capacitance of all these capacitors combined? We very well know that when the capacitor value is same, then directly we can write a capacitor in series as the individual capacitance is 10 divided by 4, we will get it as 2.5 microfarad. And this 2.5 microfarad is in parallel with this particular capacitor, that is also of 2.5 only. So we will write the net capacitance. The net capacitance will be 2.5 plus 2.5, that will be equals to 5 microfarad. So total capacitance is 5 microfarad. If you look at the options, then in 3 options the total capacitance is given as 5 microfarad. So obviously we have to go to the next step. They are also asking you the charge on each capacitor. Okay, the equivalent capacitance and the charges on each capacitor they are asking. Now please refer to it very carefully. The total charge which will flow through this particular battery will be equals to C equivalent, which is the net capacitance, multiplied by the voltage. C equivalent is given by how much? 5 multiplied by voltage is given by how much? 50. So the net particular charge will be equals to 250 microfarad. Out of that, if you look properly at C5, all the voltage is actually across C5 capacitor. All the voltage 50 volt is across C5 capacitor. So what will be the charge across C5? It will be equals to the voltage across the C5, which is nothing but 50, multiplied by the capacitance C5, which is 2.5. So if you look properly, this will come out to be 125 microcoulomb. Total was already 250 microcoulomb. Out of that, 125 microcoulomb is on this capacitor. So where will be the remaining 125 microcoulomb? That 125 microcoulomb will go in this circuit. And since all these capacitors are in series we know that all the capacitors get the equal amount of charge. So these will also get 125 microcoulomb. This will also get 125 microcoulomb. So if you look at the options, in the first option, the answer is 5 microfarad and 125 microcoulomb on all the capacitors. That will be the perfect answer. I hope this question is also clear to you. Let's move to the next. In the meter bridge experiment, they are saying that the positions of the cell E and galvanometer G are interchanged. So what they are saying is that this galvanometer comes here and this particular battery goes there. If the positions will get interchanged, then we will be left with a standard meter bridge experiment. When we discussed meter bridge experiments, we used to connect galvanometer here only, where EMF is connected, and we used to connect EMF here. So when you will interchange these positions, then also there will be no change in the null deflection. There will be a null deflection which will be occurring. And when this particular jockey will move from the left end to the right end, then obviously the ratio of R1 by R2 is to R3 by R4 will also change. Depending on that, you can get a left deflection also and you can get a right deflection also. That depends on the ratio of the resistors which is connected between them. So for this particular option, the null deflection will be obtained. Let me read the options. Only the right-sided deflection, this cannot be the answer. Only the left-sided, not possible. There will be no deflection irrespective of the position of the jockey. This is also incorrect. There will be one particular deflection when Wheatstone bridge condition will be balanced. So both right-sided and left-sided deflections, and at the balance point no deflection. Option number 4 will be the correct answer. Please don't see this option. This is not the correct option. Option number 4 will be the correct answer. Both right-sided and left-sided deflection, and at the balance point there will be no deflection. There will be a null deflection wherever Wheatstone bridge will be balanced. So I hope this question is clear to you. It was just a theoretical question. Next, the power of crane which lifts a mass of 1000 kg to a height of 20 meter in 10 seconds, and they have said that you have to take G as 9.8. This is also a simple question. Whenever we discuss power, that power is basically work done per unit time. We are talking about average power here. What is the work done? We have to just lift a 1000 kg block to a height of 20 meters. So what we'll write? mgh divided by time. Is everything given? Yes. 1000 kg is mass multiplied by. They have strictly said that you have to take G 9.8. I will not take. Why I will not take? Because the options are very far away. And let me tell you, this is 19.6 watt and this is 19.6 kilowatt. I know that I will be making an error in this question, but because the options are very far away, then to reduce my calculations, I will take G as 10 only. So M into G 10 into height. Height will be how much? Height is basically 20 meter divided by time is basically 10 seconds. Now the answer which I am getting is 20 into 10 to the power 3 watt, that is 20 kilowatt. Now obviously my answer will not match because G is given as 9.8, but I am very sure that you might be understanding here that the answer will come out to be slightly lesser than this 20 kilowatt, and the correct option, if you see properly, the correct option from here will be 19.6. To get the exact answer, you have to put G 9.8 and you have to solve. But why I will solve when I know that by approximation I can give the answer in a more better way and quickly. So this will be the correct answer. Question number 18. This again is a standard question, and it is a very simple question. Just you have to match the following, and you have to match the different formulae of different modulus which is given here on the left hand side to the right hand side, which is the formula which is given. So Young's modulus is what? We know that Young's modulus is basically longitudinal stress upon longitudinal strain. Longitudinal stress can be written as force by area into delta L by L. So what will match with Young's modulus? With Young's modulus, option number 2 will match. Now, a good student who is having a better presence of mind, he will not go further. A good student will never go further in this particular question. Why? Because if you see properly, for option number A, in only one option 2 is matching. Option number A, only fourth option is there where A is matching with 2. And I know very well that A has to match with 2. So I should not go even further. But still, because I am discussing the solution, let me tell you all the things. So first option, A is matching with 2. Then let's talk about B. So this is option number A. Then let's talk about B. Compressibility and bulk modulus. Bulk modulus is basically given by what? Bulk modulus is basically given by volumetric stress divided by volumetric strain. Volumetric stress can be written as delta P divided by delta V by V. Actually, it is negative of that. If you take bulk modulus, which option is matching with bulk modulus? We know that bulk modulus will be matching with option number 3. Okay, so bulk modulus is C option. So C option will be matching with 4. So option number 4 is C. And compressibility is reverse of bulk modulus. So compressibility will be 1 by B. So compressibility, if you see, the compressibility will be matching with option number 3. And Poisson's ratio is what? Poisson's ratio is basically given by lateral stress divided by the longitudinal strain is there and the lateral strain is there. So it is lateral strain divided by longitudinal strain. Lateral strain is basically the fractional change in the diameter divided by the longitudinal strain is the fractional change in the length. So delta D by D divided by delta L by L. So what will be the correct answer? Poisson's ratio will match with option number 1. So if you look at the options, option number 4 will be the perfect answer. So I hope these questions are clear to you. We'll meet in the next section. Now my next colleague will come and he will discuss the remaining 9 questions. Thank you.
Okay, start. Okay. So good evening, student. All of you have given today's NEET examination. So I hope you have seen the paper and many of you are getting a very good score. So I'm here to discuss the questions of physics from question number 19 onwards till 27 I will discuss. Okay. So let's take this question number 19 here. So the question tells us what, or in this question what we have, they are saying that in a concave lens, okay, they are saying in a concave lens, a ray of light emanating from the object parallel to the principal axis of the lens after refraction, what will happen? Basically in this case you can see that if I draw the diagram here, so you can see they are saying that there is a concave lens, okay, this is what we have, this is the principal axis we have, and there is an object, and from this object there is a light ray which is coming parallel, okay, so parallel light ray is coming, and that light ray after refraction is going to have what? It will have a divergence, and this diverging light ray, if you see, if I extend it backside, so it appears to be coming from this point, and this point is actually a point which is called focus point, okay. Now this is actually a principal focus, but which principal focus, okay, so this is actually is a second principal focus, okay. Conceptually it is what we have, second principal focus. So this is what we have, the ray diagram, okay. Now let's try to see what options they have given. So if we see the option number 1, so in that option they have given that after refraction from the lens, the light emerges parallel to the principal axis. You can see that, is it emerging parallel? No. So this is wrong option. Now second option they are saying that appears to diverge from the first principal focus. Okay, in this they are saying first principal focus, but you can see that it appears to be diverging from which principal focus? Second principal focus. Okay, now after that third option passes through 2f, which is the radius of curvature of the lens, so that also is wrong. Okay, absolutely wrong statement. Then fourth option passes through the second principal focus. Okay, so actually you can see that it do not passes to the second principal focus, it appears to be passes through the second principal focus. And in this particular case actually you can say no option is correct. Okay, if we go by the real concept, no option out of 4 is correct option. Okay, because conceptually it is diverging from where? Diverging from the second principal focus, but they have given first principal focus. Okay, but if I have to mark a question, appropriate answer will be what? The second option I will mark, but conceptually it is not correct. Okay, so there is a chances that NTA may give a bonus marks for this particular question. Okay, so I hope you have understood. Okay, so those who have marked second as an option, no worries. Okay, but conceptually the question is no option out of 4 is correct option. Okay, so the correct option has to be or would have been that if they would have mentioned here instead of first, what they would have mentioned? Second principal axis. Okay, or they would have mentioned here in the fourth option what? That it appears to be passing from the second principal axis, second principal focus, sorry. Okay, I hope you have understood. So next we can move. So now this 20th question you can see, in this particular question, which is a very simple question they have given, they are saying that there is a wire which is having a length L. Okay, and this wire is going to be bent in the form of ring. Okay, so this is a diagram they have given of the ring. So what we can say here, if I take the radius of this ring as r, okay, then what we can take here 2 pi r. Circumference of this ring we can take equal to what? L, and from there we can calculate the radius, which will be L by 2 pi. So that will be the radius we have for this particular ring. Okay, in this question if you see, they have given linear mass density and that is given as small m. Okay, they have given linear mass density. So what will be the mass we have? If we talk about the mass, here I am taking the mass as capital M, and that mass value will be what? It will be the linear mass density which is given by small m, and the length they have given as L. So small m into L, that will be the mass of this ring we have taken. Okay, and here you can see what they are asking. They are asking us to calculate the moment of inertia about the axis, which is y dash y. Okay, y dash axis, about this they are asking us to calculate the moment of inertia, and this axis is what we have. This axis is a tangential axis parallel to diametrical axis of this ring. Okay, and about diametrical axis we know the moment of inertia of ring we have is what? It will be m r square divided by 2. Okay, this is the moment of inertia about its diametrical about the diametrical axis of a ring we have. Okay, now what will be the moment of inertia about this axis? So that moment of inertia we can calculate by using the parallel axis theorem. So this is what we have. This is also this diametrical axis. About diametrical axis, the moment of inertia is also known as the moment of inertia about its center of mass. So that ICOM plus m into the perpendicular distance between the parallel axis, which is given here as radius. So we can take that m r square. So what will be that value? It will comes out to be that center of mass axis. What was the moment of inertia? It is m r square by 2 plus m r square, which will give you what? 3 by 2 m r square. Now the mass value we have to substitute. What will be the mass we have to substitute here? M into L, and the radius also we have to substitute as L by 2 pi. So what will be the final answer will be? It will be 3 by 2 into small m into L into this r, which is L square divided by 4 pi square. Okay, L by 2 whole square if we take, it will be L square by 4 pi square. So what we are getting now? So you can see 4 into 2, it will be 8. So 3 small m L cube divided by 8 pi square, that will be the answer, which is given where? Which is given in the first option. Okay, I hope you have understood. Okay, so I hope you have understood this question. So we can move to the next question now. So question number 21, in this particular question they have given that each side of a metallic cube of mass 5.580 kg is measured to be 9 centimeter. Keeping the significant figure in view, the density of the material of the cube can be best expressed as x into 10 to the power 3 kilogram per meter cube, where the value of x is what? So they are asking us to calculate the value of x, and that x value we have to express in significant figures. Okay, so now if you see that what we have here, we have to calculate the density. We have a cube, and we have the mass of this cube, and along with that they have given us the side length, which is 9 centimeter they have given. Okay, so if you see here, what will be the density we have? So if we write the density is equals to mass upon volume, and what will be the mass they have given? It is 5.580 kilogram divided by volume. Volume will be a cube, which is side cube, so 9.0 into 10 to the power minus 2 cube. Okay, now here see, whenever there is a multiplication and division we do, the final answer always we write in the minimum significant figures. Okay, we write in minimum significant figures. So here you can see that if I am going to solve this, the answer we are going to get is what? It is 7.654 into 10 to the power 3, okay, kg per meter cube we are going to get. Okay, so now in this case, but is this in significant figures? No. Why? Because if you carefully see here that what is the minimum significant figures we had when we are calculating the density. So you can say that the mass was having significant figures 4, and how many significant figure? This side is having 2 significant figure. So it means the final answer has to be in 2 significant figures. So here we have to eliminate what? We have to eliminate this 5. Okay, and you can see that after 5 we have 4. Okay, so if we are going to eliminate the 5 here, then this value, this 6 is going to be increased by 1. Okay, which means the final density will be what? It will be 7.7 kg per meter cube. That will be the density we have. I hope you are able to understand. Okay, so next. So question number 22, if you see, in that they have given a travelling harmonic wave equation, which is given to us 2 cos 2 pi 10t minus 0.0080x plus 0.35, where x and y are given in centimeter and t is given in second. The phase difference between oscillatory motion of 2 points separated by a distance of 0.5 meter. What will be that? So in this particular situation, you can see that between 2 points, what will be the particles which is present at 2 different points, and the separation of those points is given at 0.5 meter. What will be the phase difference between the motion of those particles? That is what we have to find. So we know the relation, how we calculate that. So that phase difference value we have is what? It will be equals to what? It will be k into delta x. Okay, where delta x is representing the distance between the 2 points, which is given 0.5 meter. Now here the k value, what will be the k value from the equation? If this 2 pi I multiply with this 0.080, that will give you what? K. Okay, because the standard equation is what? Omega t minus kx. Okay, so k value we have is what? What will be that? It is 2 pi into 0.008. Okay, and then we have to multiply delta x. Delta x value is 0.5 meter. But here you have to be careful. Why? Because 0.5 is given in meter, whereas here if you see, the x is given as what? X and y are given in centimeter. So you have to convert this 0.5 in centimeter. So what will be that 0.5? It is 0.5, okay, into 100. Okay, so when we solve this, you will get the answer. So 2 pi, this will be 100, we have 0.5, which will be coming out to be, or we can write it as what? 0.5 you can write as 1 by 2. So directly you can cancel this 2 and 2. Okay, the moment we cancel this 2, and then what we are getting here, so you can see that we will get here 0.08 and then 100. So it will comes out to be what? It will be 0.8. What we are getting now? 0.8 pi. That will be the phase difference we are getting. Okay, so that will be the answer we are getting. I hope you are able to understand. Okay, so next we can move. So again, the question number 23, which is a very simple question. In this particular question, they are saying that there is a monochromatic light which is passing through equilateral prism. Okay, so this prism which is given to us, it is a equilateral prism ABC as shown in the figure. The refracted ray QR is parallel to the base, to its base BC. The angle of incidence is given as what, 50. Then the angle of deviation value we have is what, as they have given parallel to the base, we can take the minimum deviation. So that minimum deviation is what we have, it is I plus E value will be same, so you can take it as I plus I minus A. So this will comes out to be what, 2 times of I, and I value is given as 50. So 2 into 50 minus A value is 60. Okay, so we have 100 minus 60, which will give you what, 40 degrees. So that will be the deviation we have. Okay, so deviation value is 40 degrees we have. Now after this, next question, 24th question. So in this particular question, they have given a circuit, and in that circuit they have given a AC source. Okay, and this AC source is connected to the diode and this resistor in series. This is the input which is given to us, and they are asking us to find what will be the voltage, what will be the voltage form we have across the diode. Okay, so now when we have a positive half cycle, during the positive half cycle, let's say this is positive, this is negative. So this diode is what? It is, for this diode is reverse bias. This diode is reverse bias. Reverse bias means that no current will flow through this circuit. It will be an open circuit. No current is flowing means what? That across this resistance we don't have any voltage. So entire voltage drop which we have applied from there will be available where? Across the diode. So for positive half cycle, the variation will be this. Now when there is a negative half cycle, here we can take positive, this will be negative. So this diode become forward bias. So as the diode is going to become forward bias, so the current will start flowing. Okay, and across this diode we won't have any voltage now. Okay, entire voltage we have across this resistance. So that's why we will have what? After this positive waveform, we will get what? Nothing. Okay, so the appropriate answer we have is what? It is the second one. So I hope you are able to understand. Second is the answer. Next. So now in this particular question, they have given what? We have a simple pendulum. Okay, so we have what? Simple pendulum which is having a time period T. The variation of kinetic energy with respect to time is represented by, so they have given multiple options. You can see first option, second option, third option and fourth option. We have to tell which one is the appropriate variation of a kinetic energy in simple pendulum case. Okay, so if we see the kinetic energy is what we have, it is half mv square, and we know that kinetic energy cannot be negative, it can never be negative. So straight away where we have a negative option, that will be rejected. So this second one is going to be rejected. There is no negative kinetic energy we have. Kinetic energy negative not there, so that's why this will be, second option is straight away rejected. Now understand one thing, that we know that in simple harmonic motion, if we take the displacement equation as what? If I take, there are a cos omega t, then what will be the velocity we have? It will be a omega sine omega t, or minus we have. So this, if we substitute here, so we will get what? We will get half m omega square a square sine square omega t. So the variation or the kinetic energy variation will be what? It is sinusoidal, and as we have here sine square omega t. Okay, so because of this sine square, the negative part of that sine function has to be in positive side, which means that the correct option will be this. The negative part also becomes what? Positive. So this is the variation of kinetic energy we have. Okay, so I hope you have understood this. So this will be the answer for the question. Next. So the next question they have given is what? A resistor is connected to a battery which is having a voltage, not voltage, it is having a EMF 12 volt, and internal resistance is given as 2 ohm. Okay, if the current in the circuit is given as 0.6 ampere, the terminal voltage of the battery is what? What will be the terminal voltage of the battery we have? Very simple question, straight away you can apply the relation. What is the terminal voltage formula we have? E minus I R. Okay, so E value is given, how much it is given? 12. Okay, and what is the current they have given? 0.6, and internal resistance is given 2. So 0.6 into 2 is 1.2. So 12 minus 1.2, which will be 10.8. Okay, so 10.8 volt, that will be the answer we have for this question. Okay, so I hope you have understood that. Next question. So the amount of work done to raise a mass m from the surface of earth to a height equal to the radius of earth. Okay, so now here you can see that we have what? We have a surface of earth, and this is the object we have which is present on the surface of earth, and this object from its initial position it is lifted to a point where the height is what? The height of that to which it has been lifted is what? It is equal to the radius of earth. Okay, and this will be the center, and you can take this as r. Okay, from the center if you see the total height is what we have, it is 2r. Okay, I hope you are able to understand this diagram. So now what we have to find out, they are asking us to find what will be the amount of work done we have to do. So we can apply a work energy theorem. Work done by the external agent plus work done by the gravity equals to what? Final kinetic energy minus initial kinetic energy. So here initially it was at rest, and finally also when it is being lifted to that height, we can take it at rest. So we can take the initial and final kinetic energy both as 0. So what will be the work done by external agent? It will be equals to what? Minus of work done by gravity, which will be nothing but U final minus U initial. Final potential energy minus initial potential energy. Finally, what will be the potential energy? It has minus G capital M small m divided by, if I take the mass of this particle as small m and the mass of this earth as capital M. So now divided by what? So what will be the distance we have? You can see the distance of this particle from the center we have is what? It is 2r. So we can take it as 2r and minus of minus G capital M small m divided by r. So what we are getting now? So this will become what? Positive, and if you solve this, we are getting what? Finally you can say the external agent work done we are getting is G m by 2 times of r. If I multiply it with r and divide with r, so what we are getting here? This G, m, r and r we can eliminate as what? G, or we can replace it with G. So what we are getting here? mg r by 2. That will be the work done we have. Okay, so I hope you are able to understand that how we have calculated this. Okay, so the answer will be the fourth option. Okay, so the next questions my colleague is going to discuss. Okay, so thank you very much.
Good evening, everyone. Let us discuss our next question. That is 28th number question. It is telling that an electrical heater supplies heat to a system at a rate of 100 watt. If the system performs work at a rate of 75 joules per second, then rate at which internal energy increases. See what happens that anytime when energy interactions are here, first law of thermodynamics tells us that delta Q equals to delta U plus delta W. If we take the powers, we can take time both sides as division rate, and then delta Q by delta T will tell us that the electrical heater is actually supplying some heat. Obviously in the form of power, that is what called 100 watt. It is taking delta U by delta T, and then delta W by delta T is rate at which work is executed. What it is telling that work is executed at a rate of 75 joule per second. So obviously it is 75 to be taken here, 100 to be taken here, rate at which internal energy will be actually executed or changed, that can be taken as 25 watt. So here du by dt, or time rate at which internal energy is changed, it is 25 watt. Okay, so the answer is third. We can just go on to the 29th number. They're telling that a room heater is rated at 400 watt to 20 volt. It means it is telling us some resistance of that heater. Resistance can be taken as V square by P for rated voltage square divided by power rated. So we can tell the resistance of this will be equals to 220 square, that is 220 into 220. The power of that heater is 400. So resistance can be taken as like this: 2 2s 4, 2 11s, and then 11, obviously 121 ohm is the resistance of heater. Now what they're asking that what is the power of power consumed if voltage is not this but it drops to 200 volt. anytime if applied voltage is given to us as 200 volt, obviously the power dissipated at that state will be equals to V square, which is that what was applied divided by resistance of that heater. Obviously V applied is dropped to 220, so we are taking 220 into 220 divided by resistance, that is 121. We can approximate things like 120 close to 0, obviously it will be cancelled by this, and 2 6 like this, 2 11s like this, 2 3 and 2 1 1 0. When you are solving this, obviously it will be just coming like 1 2 1 0 divided by 3. It will be approximately coming to be 331 in watts, right? That will be the right answer for this. Okay, now let us have the next question, which is, when a ruler falls vertically, 5 different persons catch it with different reaction times. What they're telling that? They're telling that a person is there and then there's a ruler there. Somehow the ruler is slipped actually, and then it falls distance h, and obviously reaction time, it means that this ruler can be caught at after some distance h, after fall of some distance h, and after consuming time t. Obviously h will be the displacement covered downside. If distance is to be taken, obviously it started from u equals to 0. If it started from u equals to 0, obviously displacement will be minus of h. It will be equals to ut plus half at square. If you take it as u equals to 0, because it is slipped from there, so taking this as 0 and acceleration as minus of g, so you will be just having the relationship of h and time, as t is the reaction time and h is the distance through which it is flipped. So obviously the time of reaction of each of the person will be directly proportional to under root of h. Now we can tell that here reaction times are directly dependent over the h. So we can tell that directly proportional to under root of h. So we can tell that here A, B, C, D, E are the 5 options over which the distances have to be taken from the reaction times. So more the reaction time, more the distance traveled, right? So we can rearrange how the reaction times are being absorbed, and then distances can be covered. So we can tell that here 0.22 seconds is actually the reaction time of the largest reaction time. That will be telling us the most or the largest distance through which the scale or the ruler have been dropped. So next will be what called 0.21. After that will be 0.20. After that will be 0.19. After that 0.18 seconds. Obviously by this order we can just rearrange it. We can have it like 0.22 seconds as the first, and then 0.21 second as the next, and at the last we can have, because they are asking us the order can be taken like let us have the last order as 0.18 seconds. That will be telling us the distances covered. So 0.22 seconds will be the first B as our option, and then 0.18 second as the C as the option. Obviously the first should be B and the last should be C. So it will be very easy for us to have the answer, because all the 3 are not having the cases as the first, or I'll say in the order is B, and last in the order as C. So here fourth will be the best option. Other can be rearranged just to have the better proper solution of that. It is just the problem of kinetic, we'll say motion in one dimension, or kinetic say kinematics in 1D. That is simply using S equals to ut plus half at square as the second kinematic motion equation, as it's a fundamental point. Okay, so let us have 31st number question. They're telling that there are 2 uncharged capacitors. So we are having 2 uncharged capacitors of identical capacity, and then these are those. C is given to us. How much C is? 200 picofarad is the capacitance, right? So here, out of which 1 is charged, so let us have the charging of 1 capacitor, and then after having, if 1 is charged by 100 volt supply, obviously V1 is given to us as 100 volt, and then it is uncharged initially. If 2 capacitors are actually having connection like this, then we know the sharing of charges have the standard result of amount of electrostatic energy actually dissipated as the energy loss in the sharing of charges can be told to us as the standard relationship is half of C1 C2 divided by C1 plus C2 into V1 minus V2 whole square. Where C1 is given to us as C, obviously second is also the same. Then we can actually treat the collection of whole the result as initial voltage supply. For the one capacitor was 100, the other was having 0. Obviously we can just calculate as filling the value of C as 200 picofarad. We can solve the rest of the part, like C equals to 200 10 raised to the minus 12 farad, right? By putting the values in the C, we can have our answer, right? Okay. So now let us have our next question. As Savita, a 11th standard student, while conducting an experiment to determine the effective length of a simple pendulum L, notes down the data of time taken to complete 30 oscillations as 60 seconds. It is asking that calculate the length of a simple pendulum actually used inside this experiment. See what is happening here. They're telling that 30 oscillations are actually using 60 seconds time. If it is the case, obviously the time period is coming as 60 by 30 seconds. It will be 2 seconds, right? If the time period is coming as 2 seconds, obviously we can know that how the length can be concluded. As a simple pendulum, we know that the length can be taken as a time period is 2 seconds, you have 2 equals to 2 pi under root of L by G, taking square both sides and L can be calculated, right? Having this as 1 equals to pi square L by G, G and pi square having the same values, L can be taken as 1 meter, okay? So this is just the question of simple harmonic motion, or you say like a simple pendulum, simple domain, which is actually using the simple data of time period of simple pendulum as 2 pi under root of L by G, okay? So obviously answer is 1 meter, simple question. The peak value of alternating current is 5 ampere. They're telling that frequency is 60 hertz. How long the current starting from 0 takes to reach the peak value? See, I know that anytime if AC is actually plotted, AC can be plotted by having the simple sinusoidal relationship like I equals to I naught sine theta, right? So it can be taken as like I equals to I naught sine omega t as the sinusoidal situation. If you are closely looking at this, they're asking that find out a time taken by this sinusoidal AC to move from bottom to peak value. So, you know, from here to here, we know that making the full wave, that can be called as time period. Obviously, they're asking that how much time will be taken in comparison to this capital T to go from here to the peak value. Tell me, what should be a time over here? We can tell this time can be taken because this is the time they're asking for making the current to go from 0 to highest value. Obviously, T can be taken as it is T by 2 and it is T by 4. The small t can be taken as T by 4. Capital T can be taken as obviously 1 by frequency, and thereby the frequency which is given to us is 60 hertz. Obviously, the answer for this time taken from 0 to highest value gathering is 1 by 240 seconds, correct? Okay, there are multiple ways to solve this also. You can also solve this question from I equals to I naught sine omega t relationship also. They're telling that time taken from going from 0 to peak value, you can fill I as I naught here and then calculate t from here also. You just do the same thing, I naught cancel. 1 will be at sine pi by 2. And from this situation, you can just have sine here, omega t will match with pi by 2. Obviously, omega t will collect the value as pi by 2 and time will be equals to pi by 2 omega. So here, if you fit omega value as 2 pi into frequency, pi will be cancelled, frequency 1 by let's say 4 times frequency will come, and then obviously that will go lead to the same expression at the last and then making the same answer as the final value, correct? So this was 33 number question. It was actually having many methods to actually resolve for the last option. Okay, so the first answer is the right answer. Okay. Now, 34 number question is telling to us that in interference and diffraction, the light energy is redistributed. If it reduces in one region producing a dark fringe, it increases in another region producing a bright fringe. See, we know that diffraction and interference are actually coming due to say the redistribution of energy which is coming through the slits onto the screen. Screen are having bright dark bands where energy is actually concentrated in the bright bands and it is actually removed from the dark bands. So what is there? It is just the energy redistribution, not the energy destruction or energy accumulation. See what happens that anytime if you are having a screen and you are having intensity a bit higher and then lower and then higher and lower and higher and lower, obviously it is just the bright dark construction. But here this is the maxima, this is minima. Here whatever the intensities are actually otherwise coming, that will be making the same straight line. But here redistribution just means that according to the conservation of energy, if any part is having lesser energy, that should go to the other parts as the increase in energy, so that total energy is conserved. Correctly stated in the first statement as there is no gain or loss of energy, these phenomena are the consistent with the principle of conservation of energy. Correct, because as whatever energy is coming through the slits, like whether it is YDSC or whether it is diffraction, single slit diffraction pattern, in all the time whenever bright dark fringes are actually coming, then the patterns are having bright and dark as just the redistribution of energy, nothing else. There is no destruction, there is no accumulation or generation, as the total energy is conserved. Obviously it is inconsistency with the first statement. Okay, now it is true, diffraction and interference are the characteristic exhibited only by light waves. See what happens that diffraction and interference are the characteristics of waves, not only light waves. Waves are starting from radio waves to last as gamma rays. So here only word is actually making thing wrong, because it is not only by light waves but starting from first wave radio waves to gamma rays. All waves are having their property as diffraction and interference. It may be told that gamma rays and X-rays and high frequency rays have lesser wave character, so they exhibit a bit less diffraction and interference. But the light waves, which are radio waves, infrared waves, microwaves, all these lesser, say, frequency and higher wavelength waves are actually having vigorous properties of waves show. So here diffraction and interference are characteristics exhibited only by light waves is actually wrong statement. Obviously this is wrong. So A is true but B is false. Yes, this is the right answer. Right? Okay, now the next is a box of mass 15 kg is kept on the floor of a stationary trolley. So we are writing here the trolley is there. This is a trolley. Now this trolley is actually to be accelerated, they're asking, and then a box is there on a rough surface inside. They're telling that yes, coefficient of static friction here mu is given to us as 0.12. Then they're asking that here, keeping the box in a stationary state in this situation, the maximum acceleration we can make this body to move, make this whole container or the trolley to move, so that it should not slide on the inner surface of the trolley. They're asking that maximum acceleration with which we can move the trolley is. So, see this box, this box does not slide if friction is having sufficient capacity to let not it to slide, right? So how to do that? I tell. What happens that when frame accelerates, then in the frame of the trolley, we can tell that whatever the pseudo force here, which is what called mass into acceleration, obviously of the frame, which will be trying this body to move in backward direction over the surface, right? What happens that if this force is a bit larger than the friction that is trying to actually make it prevented from sliding, that is what called here friction to be applied here, its highest value is called limiting friction. If limiting friction is successful in just preventing it from sliding, then no sliding occurs in the frame of trolley. Obviously, limiting friction comes here as mu s into normal reaction. Normal reaction at this point is actually weight of the body. Okay, so mu is what? 0.12. Normal reaction is what? Weight of the body. What is the weight of body? It will be 150 newtons. Okay, so now to make the equilibrium inside the frame of this whole container or trolley, we must have the mass into acceleration towards the left hand side and limiting friction towards the right hand side. 0.12 into 150 will be what called the situation. Obviously, mass is again 15, and then you will be having 15 into 12. Obviously, the acceleration, we can just tell that will be 15 and 15 cancelled. Obviously 0.1 will be going to there. Acceleration we can contain as 1.2 meters per second square. Okay. Obviously this will be our right answer. Now the next question is, 4 statements are given to us. Okay, so I think the next questions will be actually executed by our fellow colleague. Thank you very much.
Hey, good evening, everyone. Just let me know in the comment section I'm clearly visible and audible to you. Is it clearly audible? Yeah, hi. Like every year, I'm again in front of you to conclude the discussion of NEET paper. So this year, NEET 2026, after the 2025 paper, of course, many students who were reappearing for the exam, preparing for one more year, and of course, the students who entered last year into the 12th class, they also heard about the level or the quality of physics questions that came in the last year exam. Definitely, I'm sure you all have prepared. You already had prepared up to a very good quality. So in comparison to our preparation level, the level of questions that came in this year NEET exam are actually very simple. I hope you agree with the same opinion. Particularly, 2-3 problems were definitely there, in which there were some language issues like the ray optics problem, some little bit tricky significant figure problem, about some ambiguities and secondary opinions. We will definitely talk about those problems separately. Okay, but right now let us conclude our session so that you can actually get to know what are the correct answers for the problems you have appeared today. All right, so in this last session, I'll be discussing from problem number 37 to problem number 45. That is the last 9 problems for all of you. So the first problem, statement-based problem here, and they are asking you multiple statements there are, and options suggest that more than one statements given to you are correct, and more than one, like 2 of them are correct and 2 of them are wrong. So definitely you have to read all of them one by one, carefully and calmly. All right, let us start with the discussion. The first statement is the volume of a nucleus is proportional to mass number to the power 1 by 3. We all know that the radius r is directly proportional to mass number to the power 1 by 3, and as a result the volume, which is for a spherical body 4 by 3 pi r cube, directly proportional to r cube, that will come out as directly proportional to mass number to the power 1, or you can say directly proportional. So the first statement is saying volume of a nucleus is proportional to mass number to the power 1 by 3, which means it's a wrong statement. The volume of a nucleus is proportional to A, that is a correct statement. Then it comes the difference in mass of an atom and its nucleus is called the mass defect, where we already know that if we define mass defect delta M, the delta M will be given by the individual, like what is the concept of mass defect? We have the components of nucleus called nucleons, means neutrons separately, protons separately if you measure their mass, and when you combine them together in order to form the nucleus, okay, some mass disappears that converts into energy, which is responsible to bind them together. All right, I hope you can connect to the same story of binding energy. So delta M is defined as the difference in the mass of nucleons and the mass of nucleus when they are combined together. So it will be given by Z mass of proton plus A minus Z mass of neutron minus mass of nucleus. All right, that will give you the mass defect. So in the entire discussion of mass defect, binding energy, average binding energy, we are never interested in the mass of the entire atom. We are only playing with the story of nucleus. So they say the difference in mass of an atom. That's why this statement is false. Okay, and let us read the last one. Of course, we know that that is going to be true because 2 of them are supposed to be true and 2 of them are supposed to be false. The difference in mass of a nucleus and its constituents is called the mass defect. Definitely this is correct. So our correct answer is B and D are absolutely true, but A and C are false. Okay, next. The angular speed of a flywheel is increased from 600 rpm to 1200 rpm revolutions per minute in 10 seconds. So there are like two things in this problem. Most of the students attempt this problem. It's a basic problem of angular kinematics, but they have a tendency to use multiple equation in it. So first thing is we have to see how this could be done in directly one step. Second challenge here was, though a minor challenge, the game of units. One is in revolution per minute and here the time is in seconds. So we already know, I'll try to relate it with linear kinematics first, that S vector equals to U vector plus V vector by 2 into T. You can also call this as average velocity. All right. Of course, our assumption is that we are assuming it is uniformly accelerated motion case. Similarly, they are not at all mentioning anything regarding the angular acceleration in this problem. So we'll go ahead with the same assumption that we are assuming that uniform angular acceleration is there, which means first thing is how to directly take its analogous equation to solve this problem in one step. Theta will be equal to omega initial plus omega final by 2 into T or delta T, right? No multiple equations. Second thing is, as I said, units. Here it is in revolution per minute. Here the time is in seconds, but they are asking you the number of revolutions, all right? So what I'm going to do, if I'm going to find this in revolutions, this term is already in revolution per minute, then surely I should substitute it in minutes. So that minute will cancel and you will get the answer simply in revolutions. Hai na? So omega initial is 600 and be careful, if they would have said 1200 rpm in opposite sense, then I would have done 600 plus minus of 1200 because vectorially you have to deal with it now. Here, but they are not mentioning anything like that. So I'll just go ahead simply with 600 plus 1200 by 2 into time in minutes. That is 10 by 60. All right. So 0, 0 cancel, simple calculation. You get 600 plus 1200, 1800 by 2 into 6. All right. 6 will cancel 1800 at 300 times. 300 by 2 is going to be 150, which is already marked correct by our student. That's 150. So majority of these questions, especially for the students who have been connected to us from the last entire year or especially in the last few grand test papers, you could have seen that a lot of problems this year have come from the grand test papers. Of course, not the exact data, but the models. All right. And whenever we used to discuss it previously with you, we used to tell you that, you know, if something has come this time in terms of the wave optics problem, lambda by 3, then you might have done with us already with lambda by 2 or lambda by 4. So a basic variation of the data is there, but a lot of models, I'm sure you have already seen with us before. This was one of the similar straightforward problems. All right. Now let us see the next one. A submarine is designed to withstand an absolute pressure of 100 atm. I'll read the entire problem for you, but I hope you were very happy when you saw this problem in the fluids because you know, it is one of the most easiest problems which could be asked from the fluid mechanics. So what they are saying is that a submarine is designed, of course it will be fully immersed, so pressure at depth they are referring to 100 atm, which could be the maximum pressure that will be tolerated by the submarine. How deep can it go below the water surface? So below the water surface, how deep it should be there so as to tolerate maximum pressure of this much. Density and everything is given to you. So I'll just write the formula that pressure at the bottom will be equal to pressure at the top. One of the initial formula which you actually study in fluids, plus density rho g h. And what they are asking is the value of h. So that will be pressure at the bottom minus pressure at the top by rho g. Simple SI unit substitution. Pressure at the bottom is 100 atm. Pressure at the top, of course, 1 atm. So it is 100 minus 1 in terms of atm. But 1 atm is supposed to be taken as 1 into 10 to the power 5. So 10 to the power 5 pascals, SI units. Density of water, even if they don't give that, you are supposed to take it as 1000 only in SI, 1000 kg per meter cube. And G value, 10. So of course it is 100 minus 1, 99. And you will find 10 to the power 4 in the denominator, but 10 to the power 5 in the numerator. As a result, 1 0 is extra. I mean, 1 10 power is extra in the numerator. So as a result, it is 990 meters. है ना? Right. Yeah, I can see your comments. All right, all right. I hope you are understanding it properly. Yeah. Again, one of the easiest problems. 40th. Is it clearly visible for the students? Yeah, I think it's visible for you. Yeah. So the options are here on the right hand side here, left hand side. Even if something is less visible, I'll read it out for you. So you have to, like the examiner has given different components of electromagnetic spectrum and some associated property or production methods they are already mentioning in the right hand side. All right, production method of those in the right hand side. So you remember when I was teaching you EM waves, I told you that if a question comes from electromagnetic spectrum, then the rare thing is that wavelength range, frequency range, which students are always, struggling with to remember the data. Just because it has already come one time, only one time in the history of NEET. All right. But the major part to be learned and remembered from electromagnetic spectrum is their production methods, detection methods, as well as their applications. To see exactly from that discussion one problem came. Chalo, let us see how to deal with it. So microwave is directly produced in klystron valve or magnetron valve. These are actually the vacuum tubes which are used as an amplifier or oscillator to produce the electromagnetic waves, specifically lying in the region of microwaves. But I believe that generally students will not be remembering, or generally they don't read in that much depth, although we instruct you that you should be reading EM waves also. It is a theoretical topic. Every year one direct problem you get. So that theory, that much theory you should read from NCERT, points to ponder, summary. I have taken that in the last few days also for you. But anyways, suppose that you got stuck in this particular option. You should not be wasting the time in exam. Let us read further. Visible light. It is directly saying visible light, and if you see the first part in the right hand side column, electrons and atoms emit light. So when you are specifically talking about the light, then isn't it obvious that you will be talking about the visible light component of the entire electromagnetic spectrum. So because they are referring to the light here, of course, visible light must be matched with this. Gamma rays, everyone knows that is emitted in radioactive emissions, alpha decay, beta decay, gamma radiations, you already know. Infrared rays are related to the vibration of atoms and molecules. That thing you always study in material science topics also, like solids, kinetic theory of gases, and this, that the motion of the atoms and molecules will be directly dependent on their temperature, and temperature associated wave, if we talk about heat associated wave, if we talk about, that is the infrared rays. So even if you use your general logic and common sense, this problem, I would say, it was less from electromagnetic waves NCERT chapter, but more of about your common sense and general knowledge. All right. So I hope you got the correct option. Microwave A is associated with 4 and B, C, D are associated with 1, 2 and 3. A is associated with 4 and B, C, D are associated with 1, 2 and 3. I think fourth option is correct. A is associated with 4, B, C, D are with 1, 2 and 3. All right, this is code 12. If you have just joined the session, this is code 12 about which we are talking. All right, chaliye, I can see your comments. Yeah. Next, 41. Which of the following statements are correct? Whenever we are dealing with the problems based on multiple statements, our first focus should be on yes, whether they are asking correct or incorrect. Common mistakes. So right now they are asking about the correct. Inside a conductor, the electrostatic field is 0. We all know that if we talk about the properties of conductor, or if we talk about the properties of electric field lines, no electric field lines exist inside the conductor, because inside the conductor there is no electric field. Okay. If you keep some charge within the region of inside cavity of the conductor, the equal and opposite charge will be induced. As a result, whatever charge you give to any part of the conductor, that comes and resides on the outermost surface. All right. And if electric field lines, they touch or they strike the equipotential surface or metal or conducting surface, they have to do that normally or perpendicularly. All right. All these lines are NCERT lines, or you can say properties of conductor or properties of electric field lines. These are the headings or topics in which we have already taught you this much. Okay. So we'll just read all the lines for the sake of our revision and fun, that what we have remembered from our lessons. Inside a conductor, the electrostatic field is 0. That's true. Electric field at the surface of a charged conductor does not depend on its surface charge density. That's false. You have already learned the formula sigma by epsilon naught. Of course, that depends on sigma surface charge density. So that's false. The interior of a charged conductor can have no excess charge in the static situation. That's true, because whatever excess charge you provide, that induces an equal and opposite charge on the inner surface. As a result, that excess of charge is again induced on the outermost surface of the conductor. At the surface of a charged conductor, the electrostatic field must be normal to the surface at every point. That's true again. And the electrostatic potential is 0 everywhere inside a charged conductor. I will say that's false. Potential must be constant. Potential must be constant, as potential difference is 0 and electric field is 0. Potential is constant but not necessarily 0. Okay, field is 0. Potential is not necessarily 0. All right, so we will say what options are correct for us. A, C, D. These are the statements which are correct for us. A, C and D only. Okay, B was wrong. I already explained it depends. E is also wrong, because generally students do this mistake, treating it to be 0, but potential is constant. Potential difference is 0. All right, next. 42. For a metal of work function 6.6 electron volt, they are giving you a data, and basically students feel that this is a photoelectric equation based question. It's a data based question. Let us apply the formula and we can calculate. Yes, you can do. But I will again say it is based on common sense. Why it is based on common sense? You need not to touch the pen here. They are just saying which of the following wavelengths of incident radiation does not give rise to the photoelectric effect, which means out of 4, 3 will give rise, 1 will not give rise. So in order to not show the photoelectric effect, its energy should be minimum. So energy should be minimum, which means you have to choose one of those options which will possess the minimum energy, or minimum frequency, or maximum wavelength. So of course, examiner might have given this as a numerical to be solved by the students, but out of the 4 options, if you directly select on the basis of logic, you can again get the answer. That's why I said it is a very logical problem. I hope you enjoyed this type of solution. Okay, let's go on the next one. 43. A very common question in physics and chemistry, both. In the first excited state of hydrogen atom, first excited state means they are referring to n equals to 2. First excited state, that means second state. The energy of its electron is already given. Even if they would not have given this, you know all the Bohr's model and associated formula. What they are actually asking is the radial distance, what we commonly call it as radius here. So that is 0.529 n square by z in angstrom. And see, all the 4 options are in SI units. So here you have to be a bit careful. 0.529 n square, 2 square, still they are referring to hydrogen, which means atomic number is 1. Angstrom stands for 10 to the power minus 10 meter. So 0.529 into 4 into 10 to the power minus 10 meter. Of course, we know 5 4s are 20. So here 0.5 into 4 is approximately 2. It's slightly greater than that. So approximately 2.1, which anyways is common in all the 4 options, into 10 to the power minus 10 meters, which means see, option 4 is correct. All right. Don't focus much on the marking because we have taken one of the papers from our students. So definitely the marks are sometimes right. The marked option, which was previously marked, is sometimes right, but sometimes not. You focus on what I'm telling you. That's the exact key. All right. You can see what the student have marked is also quite similar. Next, 44. 2 statements are given below. When the forward bias voltage across a PN junction diode increases above a certain threshold frequency, the diode current increases significantly. So when you see a similar phenomena, not exactly the same, but a similar language used in reverse bias, you say the reverse current increases abruptly without any considerable variation in voltage. That phenomena is avalanche breakdown, and that voltage is called breakdown voltage. A similar language when we use in the forward bias, you generally call it a knee voltage. So yes, this statement is true. If we say this current is called reverse saturation current, of course this is false because initially they were referring to forward bias in statement A, whereas in statement B they are talking about the reverse saturation, which is definitely wrong. So we will say that A is true and B is false. Second option. Statement A is true and statement B is false. Easy, different situation because different type of biasing. And here comes the last problem for our exam this year. A flask contains argon and chlorine in the ratio 2:1 by mass. So as of now, their mass ratio is given, but I'll understand the story. I'll be ignoring the data for a while. Then the temperature of the mixture is again given to you. And mixture temperature means individual samples. Of course, they will be having the same temperature because they are the component of the same mixture. A ratio of the RMS speed they are asking for both of them. So what is the formula of CRMS? That is equal to under root of 3RT by M naught, where M naught is molar mass. So as you can see, this ratio what they are asking will be given by the inverse ratio, inversely related to molar mass. So inverse ratio, square of the inverse ratio of their molar mass. So I will say molar mass M naught corresponding to chlorine, molar mass for argon, which again you don't have to use the data which you have in your head. They are directly giving you the data in the question itself. You just have to substitute. So it's 70 by 40, 0, 0 cancel. Hence the final answer is under root of 7 by 2. All right. So here we conclude the discussion. And again, I'm very happy to tell you and to share it with you that this year, too many questions came from our grand test papers, which you people have already seen. And yes, the marathon sessions, which we have taken for you. So it was really a nice experience to see that a lot of problems came from that. Yeah.
Stay, sir. Hello. Sitting in my teacher's class again after completing the exam felt completely different. This time there was no fear, no pressure, no confusion, only peace, happiness and gratitude. 100%. What else we can do? And even sir, deep gratitude from my side also. Sandeep sir and please sir, please. Sandeep sir, Abhilash sir and all the team members, they just have left to home. To all the faculty members, ఎన్ని మాటలు చెప్పినా మీకు ఎన్ని చెప్పినా gratitude is a very small word. Thank you so much for educating our children. And already pre-celebrations have been started in Sri Chaitanya Gosala already అండి. ఇంతవరకు వచ్చిన డేటాస్ ప్రకారమే బొంబార్డింగ్ రిజల్ట్స్ రాబోతున్నాయి. అంటే మీకు నేను I don't say that we made question paper ఎగ్జాక్ట్లీ ఎవ్రీథింగ్ లైక్ దిస్. ఇలాంటి నేను అంటే మనం డబ్బా కొట్టుకోవడం కంటే పిల్లలు బాగా చదివారు, కష్టపడ్డారు, టీచర్లు బాగా కష్టపడ్డారు, పిల్లలకి మంచి మార్క్స్ వస్తాయి డెఫినెట్లీ. కట్ ఆఫ్ ప్రెడిక్షన్స్ ఇవి చూస్తే కనుక మీకు ఏదైతే పేపర్ ఉందో, సార్ యాజ్ యూ ప్రామిస్ టు రీయూనియన్ ఫర్ 2025 బట్ నాట్ డన్ 2025 and 2026 we are going to do dear, don't worry, because we will do it with a great victory. This year we'll do it with a big victory, right? And we are going to do it, don't worry. సో పిల్లలు ఎవరు ఎక్కువ స్ట్రెస్ తీసుకోకండి. యూ జస్ట్ ఫినిష్డ్ యువర్ ఎగ్జామ్. పేరెంట్స్ కూడా ఆన్సర్కి పెట్టుకోండి, ఒకసారి ఆన్సర్కి మొత్తాన్ని కరెక్ట్ చేసుకోండి. వాచింగ్ దిస్ లైవ్ అండ్ గెటింగ్ బ్యాక్ టు అవర్ మెమరీస్. వండర్ఫుల్. సో ఈవెన్ వీ ఫీల్ లైక్ టాకింగ్ టు యూ అండ్ వీ ఫీల్ లైక్ రీడింగ్ ది కామెంట్ బాక్స్ మోర్ అండ్ మోర్. బట్ నెవర్త్లెస్ ఇట్ ఈస్ ఆల్రెడీ 10:30 అండ్ పేరెంట్స్ అందరికీ కూడా నాదొక విన్నపం అండి. పేపర్ ఈజీగా రాలేదు, అలా అని పేపర్ టఫ్గా రాలేదు. 24లా రాలేదు, అలా అని 25లా కూడా రాలేదు. అంటే వీటన్నిటికీ మధ్యలోకి కొంచెం బెటర్గా వచ్చింది పేపర్. అంటే దీన్ని మనము ఒక లెంగ్దీ బయాలజీ మనం ఆలోచించాల్సిన చాలా ఇంపార్టెంట్ విషయం ఇక్కడ ఏంటంటే, లెంగ్దీ బయాలజీ వచ్చినప్పుడు పిల్లలు ఎలా చేయగలుగుతారు, లాంగ్ టర్మ్ పిల్లలు ఎలా చేయగలుగుతారు, ఇంటర్మీడియట్ పిల్లలు ఎలా చేస్తారు, టాప్ మెరిట్ ఎలా చేస్తారు, ఇవన్నీ చాలా వేరీ అవుతాయి. మనం జస్ట్ కట్ ఆఫ్ చెప్పడానికి ఏదో ర్యాండమ్గా కాకుండా, బికాస్ ఓన్లీ బయాలజీ ఒక్కటే మీకు కట్ ఆఫ్ ప్రెడిక్షన్కి హెల్ప్ అవుదండి. యువర్ బయాలజీ ఫాలోడ్ బై కెమిస్ట్రీ దెన్ ఫాలోడ్ బై ఫిజిక్స్. దీంట్లో ఎంతమంది పిల్లలు అయితే 3 hours టైంలో మ్యాక్సిమం క్వశ్చన్స్ అటెంప్ట్ చేశారు, మీరు చూసుకోవచ్చు. నెంబర్ ఆఫ్ అటెంప్ట్స్ చూసుకోండి. ఎంతమంది పిల్లలు అన్అటెంప్టెడ్ పెట్టుకున్నారు కొన్ని క్వశ్చన్స్, ఎంతమంది పిల్లలు కంప్లీట్ పేపర్ని కవర్ చేశారు, ఇవన్నీ కూడా చాలా జాగ్రత్తగా చూసుకొని మిగిలిన అన్ని మా ఇన్స్టిట్యూట్స్ ఏవైతే క్యాంపస్లు ఉన్నాయో అన్ని క్యాంపస్ల డేటాతోటి యాజ్ వెల్ యాజ్ రిమైనింగ్ కంట్రీలో ఉన్నటువంటి రిమైనింగ్ డేటాని కూడా ఎంతవరకు సాధ్యమవుతో అంతవరకు తీసుకొని వాటన్నిటి కంపారిజన్ ప్రకారం ఒక ప్రెడిక్షన్, అంటే ఆల్ ఇండియా అనాలిసిస్ ప్రకారం ఒక ప్రెడిక్షన్ ఏముండొచ్చు, ఎంతవరకు కట్ ఆఫ్ వెళ్ళొచ్చు, ఇవన్నీ కూడా వి విల్ టెల్ యూ. బట్ ఇప్పుడే ఎగ్జామ్ రాశారు, ఇయర్ అంతా కష్టపడ్డారు, ఆన్సర్కి ఆల్రెడీ మీ చేతికి వచ్చింది, జస్ట్ కరెక్ట్ చేసుకోండి ఆన్సర్కిని. అండ్ మీరు జస్ట్ పీస్లో ఉండండి, స్టే ఇన్ పీస్. ఎక్కువ హంగామా, ఎక్కువ ఆర్భాటం వద్దు, ప్రశాంతంగా ఉండండి. పేరెంట్స్తో టైం ఎక్కువ స్పెండ్ చేయండి. మీరు చాలా టైం పేరెంట్స్తో మిస్ అయ్యారు. మెనీ సాక్రిఫైసెస్ దే హ్యావ్ డన్. సో ప్లీజ్ ఎంజాయ్ యువర్ టైం. మీకు సీట్ రావాలి అంటే ఖచ్చితంగా వస్తుంది. సో మీకు వచ్చే సీట్ని ఎవరు ఆపలేరు, మీకు రాని దాన్ని ఎవరు ఇవ్వలేరు. ఆమె రైట్? ఇది చాలా జాగ్రత్తగా గుర్తుపెట్టుకోవాల్సిన స్టేట్మెంట్. మీకు రాసిపెట్టిన సీటు మీరు ఎగ్జామ్ ఆల్రెడీ అయిపోయింది, మీ ఓఎంఆర్ని ఎవరు చేంజ్ చేయడానికి అవకాశం లేదు. సో మీకు రావాల్సిన మార్క్స్ ఖచ్చితంగా, వచ్చే సీట్ ఖచ్చితంగా మీ చేతిలోంచి ఎవరు గ్రాప్ చేసుకోలేరండి, ఇది ఫ్యాక్ట్. సో నెక్స్ట్ థింగ్ ఏంటంటే, ఎక్కువ అనాలిసిస్లు వీటి మీద ఎక్కువ డిపెండ్ అయ్యి మీ బ్రెయిన్ ఎక్కువ పాడు చేసుకోకండి, కాస్త పీస్ఫుల్గా ఉండండి, రైట్? అండ్ ఎనీవేస్, ఆల్ ద వెరీ బెస్ట్. ఐఎమ్ very much happy that at least 2025 made many people to cry, but 2026 temporarily is making people to happy. But let's see whether our happiness is permanent or temporary for certain period of time, because once we get all the data analytics. But I'm very happy, sir, that this time both from intermediate and long term जलवा दिख जाएगा इस बार. We will show the best, because we knew it already, but it is very premature. We don't want to talk, and you will really enjoy that result. Okay, thank you so much and all the very best. Go home. If you are traveling, sir, parents ఎవరైతే ట్రావెల్ చేస్తున్నారో అందరికీ కూడా ప్లీజ్ ట్రావెల్ సేఫ్. This is very important. Do not watch the video and travel. Go home, sit, this will be there, you watch. The answer key is sent to మీకు అందరికీ కూడా ఆన్సర్ కీని పంపించాం మేము, చెక్ చేసుకోండి. బట్ ప్లీజ్ దయచేసి అందరూ జాగ్రత్తగా సేఫ్గా వెళ్ళండి. థ్యాంక్ యూ సో మచ్. బై. గుడ్ నైట్.
Bye. Good night.
చలో.